in a big way and facilitate the entire thought process, what has been generated over years by different scientific endeavors. So, with this note, I would love to yeah, please.
It's a joy to introduce Alan Lobo, Dr. Alan Lobo. He came into my life through my student Danny, who is a colleague of Alan at St. Joseph's University in Bengaluru. He said, here is a guy who wants to be associated with us and wants to work on Murburn. So Alan has been an ardent physicist, and he is into computational biophysics. He did his master's from Madhya Pradesh and PhD from Sikkim Manipal University. He is independently motivated. We took this project of, I don't know how many of you have heard the molecule kinesin. How many of you have heard kinesin? Anybody here? Kinesin? Nobody. It's Alan, do you have a video on kinesin? Wonderful. So he will take you through, and I challenged him that in the next 1.5 months, let's see if we can solve the problem. So he seems to have a solution. Let's see what it is, and let's see where he takes us. The floor is yours, Alan.
Hello. Okay, so good afternoon, everyone. Yeah. Okay. So in this work of ours, we explore Murburn concept in a slightly different way. Okay, stepping out of the influences of diffusive reactive species, we try to explore the electrostatics which governs the entire mechanism, in which not just your diffusive reactive species, but also how protein complexes and membrane complexes, they interact with each other. All right. So that's what the title is, Protein Electrostatics and Stochasticity. All right. So what is the idea? Now, when you explore the biochemistry, in which we realize that there are some complex proteins which kind of behave as mechanical machines in a very engineered sense. They have certain functionalities which are very peculiar, and they move, or they are engineered in such a way that they have a very high predictability and a very high, you can say, intelligence, and they start functioning along that intelligence. Now, normally, when we talk through physics point of view, such an intelligent engineered architecture is usually not the case at such micro scales. All right. There is something in physics which I think all the students will be very fresh with, something called principle of least action, which basically dictates that if nature has a way in which the energy requirement is lesser, nature will always use that approach as the most desired approach. That's called principle of least action. It's a very canonical principle. Canonical in the sense, a very holy principle in physics. Now, so where does Murburn concept stand there? So Murburn concept essentially challenges the intelligence aspect of these mechanical protein motors. All right. We take the example of a system in which there is a field, some force field which is happening, very general force field. And along with that, there are these diffusible charged species, what Murburn concept refers to as DRS. So they are basically charged species which are suspended in a bath, in a matrix in which they can move. And keep in mind, this matrix at that intracellular scale is a very viscous medium. All right. At that level, it would be like trying to swim through a concrete liquid, a concrete liquid. So the Reynolds number, for those of you who value the numbers, the Reynolds number is approximately 10 to the power minus 12. So it's very viscous medium. So now, if you combine these three phenomena, a field, some type of force field which is present there due to the electrostatics of the surroundings, some diffusible charged species, and a bath of mobile ions. So the system, or you can say the body, would start moving in such a fashion. This is one simple simulation which I've made in MATLAB to demonstrate what happens when a body tries to move in such a medium. All right. So what is this electromotor physiology that we are trying to talk about? The general principle, when we take the example of this protein, I'll talk about kinesin a little later. The idea is that there would be a combination of these three physical quantities: a field, the presence of diffusible reactive species, and a thermal bath, a matrix. And when these three combine together, we see phenomena occurring in a much more relaxed or passive way rather than an engineered approach. So that is the idea. That's the entire hypothesis with which we move. So we start with the understanding of this kinesin protein. All right. So kinesin is a very well-known, or you can say very well-regarded what's it called, discovery, which was done in 1985 by Ronald Whale. And he even won the Shaw Prize. He even won the Shaw Prize for this discovery. All right. This protein is known for transporting materials, by literally walking over the tubulin, which you will see in this video here, this short video here. You will see how this moves. Okay, we'll look at this video a little later as well. All right, I've shared the YouTube link here. All right, the structure of this protein you can see somewhat here. You can see these 2 feet are there. Okay, these 2 feet are there, and you can see this is the prototubulin. All right, the microtubule basically, which is composed of this alpha beta tubulin proteins. All right, you can see a lot of work has been conducted in this regard. All right, this is a well-known problem called the kinesin problem. If you just Google what is kinesin problem, this problem pops up. All right, what is the problem? The problem is, how does it move the way it does, and why does it move the way it does? All right, you will see ever since its conception in 1982, where it was hypothesized that there must be some protein which is transporting materials across this microtubule, and its discovery in 1985. A lot of work has continuously been going on to discover different types of proteins in the kinesin family and to try and figure out the structure, the mechanism in which it moves, the structural dynamics in which it moves. That's the most recent topic in this field of kinesin proteins that is going on. All right. So the question is not whether it moves or not. We know, we have experimental evidence also that it actually moves. But the question is, how does it move the way it does, and why does it move the way it does? All right. That's the point of focus. Now, what is the classical story behind it? The classical story is that an ATP docks to one of the feet, which is actually called the kinesin motor domain here. These 2 feet you can see here. The literature refers to these 2 feet as kinesin motor domain. So that is what we'll call it throughout the presentation. So the classical idea is that ATP binds to this feet and somehow gives it an energy to jump by a very well-defined distance of 8 nanometers and then move to the next. All right, because of this is how the motion actually looks like. So if you look at the 2 feet here, you realize it's a very structured, very engineered type of motion. All right, and this is the classical idea behind this. Okay, this is the classical explanation behind this. But this classical explanation brings with it a lot of questions. First question is, how does the protein know? How do the 2 kinetin feet know exactly where to step and how to step? And how do they know of this exact 8 nanometer step? All right. How do they know of the gait, the pattern in which they are walking? The pattern that is visible in this video is called hand over hand pattern. All right. In which they are actually, walking like we humans do. So how does the system know? And second thing, as I told you, the system is actually a very viscous medium. So systems cannot just walk very freely as is represented in this animation. In reality, you require a strong force for the protein to move, for the complex to move here. So where does this force come from? Is ATP alone sufficient? When we look at the experimental evidences, we realize that some numbers do not match. So now let's take a look at what is the experimental observations. First of all, as I told you, each step is 8 nanometers, exact 8 nanometers, all right, which is a very well-defined value. We have seen this in cryogenic photographies. And also, the maximum speed with which it can move is 800 nanometers per second. All right. And it can move a maximum of 1 micrometer in one run when it is unloaded. That is when the kinesin is unloaded. If you look at this big vesicle here, this is the load inside which the stuff is being carried. Now, what are the problems with the existing literature? There are a lot of problems actually. All right, the possible explanations. First of all, there is no explanation of why it moves the way it does. Second, there is a lot of confusion of the pattern and the symmetry in which it moves. All right, you can say the hand over hand fashion, or there is an asymmetric walk. Sometimes there is an inchworm fashion in which one hand, or you can say one feet, moves more than the other feet. So there are a lot of questions regarding this. One major problem is the presence of intelligence. So how does this protein complex know about the exact values? All right, this 8 nanometer step and stuff like that. All right. So what is the hypothesis derived from the Murburn concept? Of course, Murburn concept tries to challenge this molecular intelligence idea. All right. So as I told you, Murburn concept, apart from the actions of DRS species, diffusive reactive species, it also has another, you can say, a more global outcome, this concept. The outcome is stochasticity plus the interaction of diffusive charge species. Now, the moment you say charge species, you realize you're dealing with electrostatics. All right. And the moment you say electrostatics, coupled with stochasticity. So stochasticity is actually a very important term here. Okay. It means motion of bodies under the influence of some random noise. All right. So what does it actually look like? You see the simulation here. You see this, the blue particles are the particles which are the matrix. They are continuously colliding with this red particle here. And under that influence, this red particle is moving randomly. This is called Brownian motion. This is the most fundamental example of a stochastic model. All right. So this type of a behavior coupled with electrostatics is what Murburn concept actually presents in terms of physics. So when you couple these two together, you actually create a new, you can say, field of study in physics, soft matter physics, as a matter of fact, electrostochastic thermodynamics. All right. And we have models, we have mathematical models which kind of explain such systems. All right. So as the very beginning of our work, what we try to do is we try to create a mathematical model to replicate a system moving in a media with the same parameters as our cellular matrix. All right. We chose the Reynolds number as 10 power minus 12, which is an experimental value. We chose the temperature to be 300 Kelvin. All right. The unit of energy is 1 kBT, where kB is the Boltzmann constant, stuff like that. So the equation and all this is the Langevin equation. We'll not go into too much detail of the equation. The results are what is very interesting. All right. So what are the results here? I will tell you. First of all, there are experimental data which tells that to move at that distance, to move at that scale, the protein requires an energy value of somewhere between 12 to 16 kBT. All right, 12 to 16 kBT. To put in perspective, 1 electron volt is equivalent to 11,600 kBT. All right, so that is a scale that you can refer to. Now, when we do not use this idea of a power stroke, that is the ATP giving some sort of a spring to the feet and jumping from one point to the other, we are still able to calculate this energy value very precisely. That is 13.5 kBT. That is, if we just allow the system to move by itself in this media, we still see that the energy requirement is the same. Also, what is interesting is that the force that was required, the experimental evidence says that the stall force of 6 to 8 piconewton is required, and we get the calculation to somewhere about 7 piconewton. All right. And also the maximum speed with which the particle can move, the system can move in this type of media is 800 nanometers per second, which is also the experimentally derived value. What this means, now you can see a lot of resemblance here. It does not conclusively prove that maybe the stochastic model is correct, but it also points us to the right direction. At the very least, it points us to the right direction. What it also does is that it shows that the hydrolysis of ATP, which is what is responsible for, in the classical model, to cause this feed to move at that distance, is the energy equivalent of one ATP hydrolysis is 1 kBT. All right, 1 kBT, which is mentioned here. However, in order to move by that distance, the minimum energy required is about 20 kBT. So there is a huge mismatch in these values. So now the question is not only whether ATP is responsible for it to move or not, but the main question is, where is this extra energy coming from? All right. So now we go back to the Murburn idea. We try to find if there is any electrostatic interaction between the kinesin feet and the tubulin on which it works. So we do a lot of, what's it called, studies on the protein structures of the concerned systems here. So this here is an alpha beta tubulin, all right, which is the structural unit of one protofilament that is in the microtubule. And what we do, we create a path using this alpha beta tubulin like a cylinder. You can see this cross section. Now the idea is that the kinesin starts to walk on top of this cylinder, right? The kinesin will walk on top of the cylinder. If you look at the video, if you see that video once again, you will realize that this kinesin is actually, you can see this kinesin is walking on top of this. So each structure here, each structural component is this alpha beta tubulin. All right. So we are going to analyze the feet and we are going to analyze the path on which it works. So what I do is using this workflow. All right. I take the PDB, I clean it in Chimera X, and then I convert the PDB to PQR, that is the coordinate file set of all the atoms in the protein complex, and I pass it through APBS, which is basically a Poisson-Boltzmann solver, which gives me the potential, electrostatic potential landscape. All right. When that happens, what you see here is that the electric field and potential of the region around this alpha beta tubulin is in this graph, the electric field and the electric potential. Above is the electric potential, below is the electric field here. All right. Now this alone does not make a lot of sense here, okay? Because there is not just one dimer on which it works. So what I do now, this is the actual protofilament. As you can see here, the actual cross section of a microtubule. The PDB ID is 5SYF. It's taken from RCSB. Now I take two sections here. One, as you can see, along the surface of the microtubule, and the other along the axis of the microtubule. All right. Now it is known that, as you can see here, the kinesin will walk on this surface here, as you can see, this rod-like structure here. All right. So what I do is, and I analyze the electric potential and electric field, electrostatic potential, electrostatic field around this protofilament. Or when I do that, I realize something. You see a lot of periodicity here. All right. When you look at colors, as it becomes red, it is more positively charged. As it becomes blue, it is more negatively charged. You see a lot of periodicity. All right. That periodicity dictates something. It means that the potential field is actually repeating itself. All right. So you can see that same thing here. So this is just a cylindrical version of the same protofilament here. All right. You see a lot of periodicity. What is very interesting is that the distance between one high and one low, that is one peak positive potential and one negative potential, is exactly 41 angstrom, which is 4.1 nanometer, which means the distance between every two positive potentials is exactly 8.2 nanometers. All right, which is very much in lines with the step size of this kinesin heads. Now I look at the three heads here. All right, the kinesin motor domains, kinesin feet here. So we have the apostate. Apostate is the state where nothing is attached to the feet. And we have the ADP attached feet and the ATP attached feet, ATP-like attached feet here. And what I do is, now I know that the protofilament on which this feet, this kinesin is walking, is actually a charged system. It's a landscape. It's a landscape of electrostatic potential. So now what interacts with a charged field? A charged body, right? So if my hypothesis is correct, the three heads should be charged, should have some charge, and they do, as a matter of fact. All right, so this is the potential of the charge landscape of the kinesin feet in the ADP state, ATP-like state, and the apostate. All right, so you see here, it's not just that they are charged. You see the local concentrations of positive charges and negative charges in certain regions, which means the feet, they actually behave like dipoles. So now what I do is I take this feed and I just carry it and move it along the protofilament just to see how this feed interacts with the potential of the protofilament here. When I do that, I realize something very interesting. When you look at this graph, you see that when ADP is attached to the feed, this blue line here, it is rather free as compared to the APO and the ATP state. What it means in biochemistry, it's basically when you have this protofilament and you have the feed of the kinesin, if the kinesin feed has ATP attached to it or nothing attached to it, in either case, it is very nicely fixed on the protofilament. But the moment ATP hydrolyzes to ADP, it becomes loose. You see this energy gap here. The y-axis is energy gap. You can see a huge and a kind of a constant energy gap here. So you see, because of this, because of the ATP hydrolysis, ATP becoming to ADP, that is, the feed becomes loose. All right. So once the feed becomes loose, there must be some force which causes it to move in either way. All right. So we'll see that as well. So after this analysis, what we do is we perform a steered molecular dynamics of this entire study. And when we do that, we again realize something very interesting here. As shown, now this is actual data. This is not a mathematical model, but this is actual molecular dynamics being run on the cryo-EM structures of the protein heads and the protofilament. What we see is ATP, the blue dots and the black dots are ATP and APO states respectively, and the red one is the ATP state. So what is happening here is when you take this protofilament and the kinesin feed with APO state or ATP state, it is very closely attached, as I told previously. When this ATP hydrolyzes to ADP, what happens? It becomes loose. So it becomes loose, it becomes detached. Now, when that happens, you see the slope of potential field of the ADP state. See here. The slope of the, you can say, incoming potential and the slope of the exiting potential is different. Now, let's not go too much into physics. The idea is that force is actually the gradient. It depends on the gradient of potential energy. So the very fact that the slope is different means that there is a forward push. There is a forward push of the, what's it called, electrostatic force on this hydrolyzed kinesin feed plus ADP, which pushes it, pushes it forward. So the complete, cycle or the walking cycle results in this mechanism. So we have two APO feeds. All right. Now what happens here in the back feed? ATP attaches. All right. Now when ATP attaches, then it hydrolyzes to ADP. The moment ATP gets hydrolyzed to ADP, the feed leaves the protofilament. The moment it leaves the protofilament, there is an asymmetry in the forward and backward force because of which the ADP swung forward. It swings forward. All right. And if you looked at this plot, you see the first binding site is 2 nanometer and the exact next binding site is 10 nanometer. So these are the most favorable positions, electrostatically speaking. So that is why the step size is exactly 8 nanometer. All right. So as it swings, then again the ADP is lost and the APO feed goes forward, and then this previous feed again, goes through the same cycle. All right. So this is how the kinesin walking cycle can be explained. All right, using computational data in this case, using the Murburn concept. So what all things have we been able to explain? All right, which were observed in the cryogenic experiments. The 8 nanometer step conserved, the 8 nanometer step size. This has been explained very nicely because of the periodicity of the potential in the protofilament, that is in the microtubule surface. The directionality, it depends on the potential landscape of the ADP added kinesin motor domain. Also, there is no need for an engineered power stroke anymore. ATP to ADP hydrolysis does not cause the feed to jump from one point to the other. It merely detaches the feed. The motion is happening because of the electrostatic interaction. All right, according to the Murburn model. All right. Now, one more thing which is very interesting here. Now, the gliding part, what I mean to say is that this electrostatic force is a conservative force. What it means that if A exerts a force on B, B will exert a reaction force on A. What it means is that now, suppose, hypothetically, if I hold this kinesin at its position, the protofilament should start to move backwards. All right, which has been actually observed experimentally. All right, sounds very weird, but it has been actually observed. And the symmetry of how it takes these steps is once again dependent on the potential landscape of the protofilament. As a matter of fact, it is these asymmetries because of which other proteins in the kinesin family, that is kinesin 5 and all, they are even able to move backwards. All right, not just forward. So, all right. So now this approach explains the interactions of protein complexes, but it also opens an avenue in which how externally applied charge species and electrostatic fields can be potentially used to interact with biochemical systems, and potential applications can be explored. So such is the interaction of whole plasma. So this is a very engaging field which many biophysicists and biochemists are actually working on, and has a lot of potential applications. So, okay, so thank you.
Congratulations for very good work.
Thank you.
There are two aspects of the physics involved in this system. One is, as Roger Penrose defined, the microtubulin, the quantum system. It's a quantum mechanics system.
Yes.
And therefore, there are a lot of quantum mechanical
calculations.
Yes, sir. Necessary for that. That is in one scale. In another scale, the whole system is an open system, open system.
Yes, sir.
So that thermodynamics which
yes,
is used is not a thermodynamics which we usually use in our
yes,
we have to use a thermodynamics in the far equilibrium system,
yes, sir.
which was elaborated by Ilya Prigozhin.
Yes, sir.
And how chaos sets in such a system. Therefore, what I feel is that this is a very beautiful concept, Murburn concept. It combines the Ilya Prigozhin chaotic system to Roger Penrose quantum system.
Yes.
And therefore, we may be able to come up with a new mathematical equations.
Yes.
For example, when we analyze that that electrostatic potential as it moves, and if we just calculate the information entropy,
yes, sir.
K2,
yes,
K2 entropy, that's a beautiful concept where we will see it's not a stochastic process.
Yes, it's not.
This is not a stochastic process.
Correct.
It is a outwardly seen stochastic process.
Yes.
It's a deterministic process.
Yes, sir.
And you can actually find out the D2, that
diffusion coefficient. Yes, sir.
I have second order dimension.
Yes, sir. Yes,
second order dimension from that concept, and that will clearly give. And also it's a nonlinear language equation, right?
Yes.
So it's a very beautiful concept. Lot of work can be done with this. Simulate, predict, and the concept. Beautiful work. Congratulations.
Thank you, sir. Thank you. So what sir was saying, I would like to say something. So what sir was saying is the coupling of quantum systems at a very small scale, angstrom level scale, and a little bit larger if we go to micrometers or tens of nanometer scales. So this model then definitely becomes something called a semi-classical model. So what sir is referring to, the Langevin equation, this is that equation that sir is referring to. And the exact diffusion D1, D2 terms that sir has asked to calculate, we have actually calculated. And they really seem to be fitted within the experimentally observed data. So it agrees with that semi-classical, part of which is quantum, part of which is a classical model. Yes.
I wanted to say something here. This is the first time that, in the last 27 odd years, I've been working with collaborators and all that. This is the first time that I am not solving the problem. I suggested the problem, and a collaborator, I really don't know what's really happened. Now I have to sit with it. He took the problem, something like, I said the problem, I said, these are the things. Would you take it? Would you do it? Do you want me to get into it? No, I'll take it. That's what he said. I still don't know what he has done, how he has done it. But then this is the way I hope Murburn goes forward, in the sense that I don't want to be solving everything. People should join in, people should take it forward. Because now I think with Murburn, I'm coming to the end of, I want to get into social work. So I've solved enough number of problems. So if Professor Nampoori says that it's good, I trust him. And he said something to me yesterday about this work. I was so sleepy, I haven't slept for so many days. I will sit and listen to him more. Thank you. Yeah.
Thanks.
There's a beautiful book by Roger Penrose, Shadow of Mind.
Okay.
Shadow of Mind. But all these descriptions are there. Quantum system.
Oh.
Dublin as a quantum system.
Actually, that was what I was also saying to sir in the previous session. Actually, the quantum explains it better.
Yes.
Than the stochastic process.
Yes.
I'm a biologist. I don't know much about the
No, sir, you are right.
physics. So that is what now sir is also saying, because I was working with anesthesia with the Penrose model. Penrose model, there is a model called a Penrose hammer of model for anesthesia. So that beautifully explains how the anesthetic is working.
Yes.
Really contrast of the current concept in
Yes.
anesthesiology.
So in that line, sir, actually this equation is a classical equation, but I solved it quantum mechanically. So what you see here, position, is not position, it's the expectation value of rate of change of position. So we use wave functions and then we solve it. If I showed it, it becomes a little complicated. So we skip it in the analytical pathways. NLSE, yes, sir. Thank you, sir, for mentioning.
Thank you for the nice presentation. It's not 100% stochastic model, as Nampoori sir said.
Yes.
Because you have alpha, beta, tubulin heterodimers repeating every alternate segment.
Yes, sir.
And you see that your, I think the next slide or one of them, where there are so many interfaces, regularly coming.
Yes, sir.
See, you can see the blues and the reds.
Yes, sir.
So which mean that there is some determination,
Yes,
or there is some traffic is going on in that direction. But how do you consider the entire thing as one single unit? Say, for example, you have a cargo sitting on top.
Yes, sir.
And you have the cargo is bigger than, I always equate this to നമ്മുടെ നാട്ടിലെ പുളിയുറുമ്പുകൾ വലിയ മാവിന്റെ ഇലയും പിടിച്ചുകൊണ്ട് ഇങ്ങനെ ഇതിന്റെ മുകളിൽ കൂടെ പോകും. So it's like that.
Yes.
So how does it so wonderfully does it? So the entire thing is working as a unit. So when it works as a unit, so our perception of, because we saw the beautiful video where you only seeing part of the whole story.
Yes.
But surrounding that is where the Murburn is playing a big role, where all these electrostatic interactions and the oxidative reductive process, etc., is helping to maintain the whole thing so that the rest of the people can do its job walking over it.
Correct.
So that way it's very interesting. So you may have to do a little more of a work along this line so that it can be made a lot more better.
So the motion of the final step, that is the, so there are two things that I've done here. In this case, I've actually taken this head and I've dragged it along manually to see how it interacts with the potential. But this just gives me a hint of what is supposed to happen. The actual molecular dynamics run, this is done by solvating. So it's a 15 by 15. So there's a lot of water molecules also. And for neutralization, we add 150 millimolar KCl solution. So what you're saying is exactly correct, sir. So it's a big thermal bath. A lot of things are involved. And collectively, this type of a pattern is coming. And the only reason this is very noisy, you can see a lot of deviation, is because I pulled it rather quickly to finish it the given timeline. But I'm still running it. The second thing is that protein, that cargo problem. What is known right now is if the cargo load exceeds the given value, it starts moving backwards. So according to these results, the hypothesis would be that something fluctuates in the potential landscape. But for that, now we have to start increasing the load. So that is the next part.
So that's the thing. The cargo load, if the potential force is enough to pull the cargo load.
That's what, sir. Yeah.
ഒരു കാറിന്റെ മുകളിൽ ലോറിക്ക് വിൽക്കുന്ന സാധനങ്ങൾ വെച്ച് കഴിഞ്ഞാൽ എങ്ങനെ കാർ ഓടിക്ക് വിടത്തില്ല. So how do you bring that aspect into it? Because so you have to consider the entire thing as a one single unit, and there is a unidirectionality in here, and how these potential differences are playing a big role, in terms of, because some of the things what you showed is very interesting.
Yes, sir.
And there the alpha beta tubulin heterodimer repeating
Yes, sir.
aspect is coming in a big way.
Yes,
So it's
sir.
a very organized system, but at the same time, there's a lot of chaos in there.
Yes, sir. So it's a deterministic but a stochastic system,
So-
not non-deterministic.
Yeah.
But the idea of or the concept of Murburn plays a role in the electrostatic landscape. It's a bigger picture overall, and definitely so that systems, that what you're saying, the cargo, considering the cargo as a part of it, becomes a problem of the systems biology, which would be a much bigger computational work. But that is the next step.
That
Yes.
is one issue with the problem with the computational
Yes.
modeling work, because you can't recreate
Everything.
entirely a
Yes.
cellular combinatorial complexity
Yes.
in that system.
Is there a parameter which you can control over this whole? system?
Yes, sir. So
That parameter
yes, sir. That is there in each stage of the calculation. So if I
is a
start,
control parameter, yeah.
if I start from the beginning, so Reynolds number, the diffusion
Yeah.
constant, temperature.
There is one control.
Yes, sir.
So, by changing that control parameter,
Yes, sir.
then we may be able to come up with a
Yes.
period doubling system.
Yes,
Period doubling system.
yes,
And ultimately it goes to chaos.
yes, sir.
Inside the chaos, there will be order.
Yes, sir.
So life is that to be in order.
Only in a small
Yeah.
scale. Yes,
Is that emergent out of that chaos.
yes. Thank you, sir. Yes, sir.
I would like to make a comment.
Yes, sir.
Chemists are always interested in molecular motions.
Yes, sir.
Indeed, I think in 2016 or 17, Savage or Stoddard, etc., got a Nobel Prize for molecular machines. 2016 or 2000, I don't remember exactly. But what happens is that they thought about fulcrums, molecular motions, etc., based upon conformational
Yes, sir.
changes. Now, the interesting aspect I wanted to just think here is that in this we can perceive what you have shown as a video as a molecular motion,
Yes, sir.
X moving on Y.
Yes, sir.
Okay.
Yes, sir.
So now what happens is that this molecular motion is not only simply X moving on Y, but here in reality there is a solvent system,
Yes, sir.
as well as there is weak intermolecular interactions.
Yes, sir.
The moment when molecules are moving,
Yes, sir.
they are interacting with each other.
Yes, sir.
So where does these two concepts, that is
Yes.
the solvent aspect,
Yes, sir.
plus the very weak intermolecular aspect taken care in your simulation
Yes.
or calculation?
Yes, sir.
This is the
Question.
the comment is our chemist interest in molecular motion.
Yes.
The question is these two aspects.
Yes. So the answer is, first, when I manually drag it, there I do not consider these things.
Okay.
But when I perform the steered molecular dynamics on GROMACS, it is engineered in such a way that it has to take all these effects, all the small to big forces, all the small to big interactions with it. The idea is, if this picture gives the same answer as the previous picture, then the small interactions that you have just discussed about are either too weak to be considered, or they are, as a matter of fact, enhancing the effects. So we see that here actually. So we see that exact periodicity of exact 8 nanometers occurring because of the combined effect, not just the electrostatics, but all the weak forces, the van der Waals forces, everything that is present here. So the molecular dynamics, I don't have to solve any calculation. I have to give the system, solvate it with the, what's it called, all the water and everything that is there.
So the embedded algorithm will take care of it.
Yes, sir.
Okay.
Yes, that's right.
But it is what is really profound is that the energy to push.
Yes, sir.
Okay, the petrol is the difference in the slope.
Exactly. Yes, sir.
In a formal way of saying,
Yes, sir.
difference in the slope is the pushing.
Push. Yes,
That's
sir.
very, very interesting.
Yes.
Fantastic.
Thank you, sir.
Yeah, I mean, the younger generation should ask a lot of questions, because these are all interesting things as we speak. This is happening in our system. There's so much of axonal. This is actually called anterograde axonal transport, and where the kinesin is involved in the whole process. And requires a whole lot of ATP energy involvement for the movement of this unidirectional process. So it's very interesting actually, because in diseases like Alzheimer's disease, many neurodegenerative disorders, all this has been affected. And there is so much evidence which we ourselves have created across the world. And the drugs are targeting these phosphorylation events, in which otherwise called as energizing events that is taking place. So I think you guys should actively involve in all these processes. It's a very interesting thing. In fact, some of the things can solve a lot of disease problems. Thank you. If there is no question, we'll go to the next.
To say something here. What's happening here? Yes. Who's it? Yeah. Sendil.
Yes.
How that's explained in your
no, that I have removed that slide actually.
okay.
But we saw that. Okay, so this was one aspect. This work extends beyond this. So what I do is on the same protofilament, now I keep two heads, and I try to see how, in using molecular dynamics only, I try to see how they're actually moving. And I also showed here that the feet, they are not just charged bodies. If you see here, they are dipoles. Dipoles means they will not just move in the electric field. They will move while rotating in the electric field. All right. So that hand over hand swinging motion, no, that happens because of this dipole pattern. You see here, I'll just explain how is it. It's like a polarized molecule. Okay. So here you see this is a negative charge. This negative is blue. Just the color convention, I've kept it that way. Positive is red. Okay. So you see in the ATP state and the apo state, there is more differentiation of the red and blue. All right. Whereas it is not so much in the ADP. You can see the distribution is kind of, so it might not be very clear by looking at these plots. But when you actually calculate the dipole moments, you see a substantial difference. That is actually the reason why ADP is released.
So you're saying one might be ATP-like and one might be apo. Like one of the legs is apo and one of the legs is...
No, I've explained that here, the mechanism. All right.
Okay.
So this mechanism itself dictates that the jump would be symmetric. Now the question is, at what case? The question is not whether it is symmetric or asymmetric. The question is, in which scenario will it be symmetric? In which scenario will it be asymmetric?
Yes, I understand that. What my question was, so let's say apo-like and then you had ATP-like also, I think.
Yeah.
Go back to that slide. So you have two cases. So there are two different...
three actually.
Three. So each of them have a separate distance which they can move. So for example...
No.
So all of them have the same 8 nanometer...
I'll explain. Okay. So the apo and ATP versions are very strongly interacting with the present potential, because of which they are very closely attached. The electrostatic pull between them is very, very nicely. Okay, think of a ball. All right. Now on the ball, there is uniform distribution of positive and negative charges. Right. What is the total charge of the ball? Zero. Right. But in the same ball, suppose on one hemisphere, you have all the positive charges, and on the other hemisphere, you have all the negative charges. Total charge is still zero. But isn't that polarized now? Which means now if you keep that on the potential landscape, it will rotate. Did you understand?
Yes.
So that type of interaction is what is happening here. All right. So in these two cases, there is strong interaction, whereas in this case, it is weaker interaction. All right. So actually, Abhinav sir helped us last night with a lot of dancing to actually explain how this phenomena happens. We were discussing all this till I think 2 a.m. So what happens is when now suppose you have this apo. Apo means nothing is attached basically. Okay, that's why I use this term apo because the literature, the PDB-IDs use the term apo. Okay? So you have this feed in the apo state attached on the protofilament. Now ATP attached to it. So now if you look at the values of the energy, you realize in the black and the blue, there is not much difference, right? Which means the structural strength or the gluing strength does not change when AP converts to ATP, T. But when ATP hydrolyzes to ADP, it becomes loose. All right. See, the energy difference is positive, which means now ADP state has more kinetic energy. More kinetic energy means it's free, like a free electron, right? When that happens, it becomes loose. The moment it becomes loose, it will start experiencing this force you. Even the ATP is experiencing the force, but because it is so nicely attached, it is not moving. But ADP is weakly attached, so it's pushing. That is what it is.
Thank you.
Yeah, I just wanted to say the things that we are presenting here, some of these things, they are not published. We are bold. We don't care. We are sharing with you. If you do real cutting edge works, you just go on. All right. So yeah, thank you. I think. Yeah, I think. Thank you.
Yes,
thank you.
Thank you. Thank you.
Put your hands together for. We have a small issue. The next speaker was Abhijit Anandakrishnan. He has been summoned by the Institute Administration to Kollam. So I'm taking over because nobody else. Hello. Yeah. Good afternoon. Can you put my slides on, please? Where do I see? I have to see it like this, right? Okay, I'll come here. That's much better. Sorry. There you go. Can you enlarge it, please? Well, okay. Okay. So I'm going to talk about vision, which is also photoreception. This is a pretty long talk, but since you're famished, you're hungry, and sir also wants to leave quickly. He has some other function elsewhere. What I'll do is I'll try to focus on the critical aspects. Now, I am going to present with the intention of exciting only the younger people here. All right. So I request you people to play along. This is now this part. We'll just try to show you certain things that you might have seen in your textbooks while you studied, but you did not ask certain fundamental questions. Okay. Now this is sun. This is the structure of an eye, human eye. You see here, this part is enlarged, and in the schematic, you see it here. So there are different kinds of cells, and this part is the rods and cones. All right. And then you have, if this is the retina, you have blood circulation here, blood here, and then the nerves, they arise from the rods and cones here, and they come all the way and go into the optic nerve. All right. So in the classical theory, rods and cones are deemed as the fundamental units of vision. Now, basic chemistry is that, this is the retinal cycle. It's been given a Nobel. So now photon falls. This is the active photoactive chemical in the protein called rhodopsin. Sorry, my bad. Rhodopsin. Photon makes cis to trans. All right. And then this cis to trans movement or the photo-induced change gets a lot of things going. I'm not going to go into the details because that's beyond. So, and then it pops out, and that sends a signal to the protein that's adjoining rhodopsin on the membrane. And then that leads to, that's called, it's a, GPCR, transducin. It's a protein, GDP binding protein. So it leads to activation and conformation change. One thing that biologists are very excited always about, when they don't understand, they always invoke conformation change. Yeah, well, I don't say that it doesn't exist. Definitely it does. But then not every signal transduction, not every receptor ligand interaction leads to a conformation change. Certain things you cannot explain otherwise also. Then there is phosphodiesterase, which leads to a cyclic GMP cascade. Now, this is how a rod cell would look like when looked closely in conjunction with the retinal pigment epithelium. So it has mitochondria, it has got the synapse, then there are this disc. This is called the outer segment, all right? So let's, and then the resting and excited states are characterized where there are different types of the resting state is like this, and the activated state is photon trans, it becomes, then GDP, GTP is bound, and then the calcium ion channels and cyclic GMP cascades and all are activated. I will try to just give you a brief view of what it looks like in the classical perspective. Photon hits, it jumps out, it somehow triggers a GPCR activation, transducin, somehow the alpha transducin breaks free, goes, first what happens is the GDP that is bound, it gives way to GTP. GTP is preferred. And then the alpha transducin breaks free and goes and binds to phosphodiesterase, and there is a signal cascade. So the rationale of exchange of GDP with GTP is the key signal transduction by GPCR or transducin. All right. So now this is the part that I want all of you to focus. How do you do something new? You do something new when you question something that is old. All right. That's where your relevance comes. If you have seen a wonderful movie called Robots, it's a Pixar movie. There's a character called Oscar Wilde. He says, find a gap, fill the gap. That's where you come in, whether a scientist or an engineer. Scientist explains a natural phenomena, engineer solves a problem that mankind faces. So you need to find a gap and fill the gap. So now I look at the problem like this. When I was your age, I had seen it multiple times, but then here is the problem. This is the structure of the eye. You see multiple layers, approximately you see. Now you see this so many layers of different components, many of which are active. Blood flows in, activity, cells, components have colloidal movements, all that. So I'm not going to go into the details of the anatomy. The point is this, that the signal should rise somewhere here. It should travel all the way and then go like this. This is the action potential. Now, I do not understand why, if this were the case, why didn't we have a simple system where the rhodopsin was, the rod cells were facing this way, and you had a simpler system. You just need a diffuser. The classical theory says that how our eyesight evolved to take care of diffuse light. If that is the case, just a diffuser. It need not go through this. The problem is, roughly you say 350 to 400 nanometers, microns is the thickness of the retina. Now, a photon of 400 nanometer wavelength, the shortest one, if it has to traverse 400 micron, it has to traverse how much times the wavelength? 1,000 times. So suppose a photon arises here, comes here, goes here. Now here, it will go through, before traversing this, it will go through reflection, refraction, diffraction, all the different kinds of processes. What is there to prevent it from happening? So if you get some signal like that and it falls on retina, a photon comes from here. Once it will come here, it will go here with a different Q, and sometimes it might come here with a different Q. How could you possibly ever evolve a photoreception system with this kind of a setup where rods and cones are the primary phototransducer? So that is the fundamental question that I asked. Then the structures of the protein involved. Now this is the rhodopsin, this is the transducin complex. Now rhodopsin is not, see, how can we explain conformational changes mediated? What is the beta transducin's role? This is alpha transducin, this is beta, then is our omega. There is no role whatsoever for this cyclic kind of a protein there. All right, this is the alpha transducin. The point is both GDP and GTP bound alpha transducin structures are known. There is no real conformation change. It's practically one lying over the other. Hardly any difference. So how does the classical rationale get explained? So now there is no role in the classical theory for oxygen in vision. Yet we find retina, both sides are highly innervated by blood cells, blood vessels. So why do we have blackouts in hypoxia, proximal and distal surface vasculation? Higher light gives more ROS production. Why should greater amount of light give more ROS? Photo bleaching at high intensity. Then the classical cycle, the retinal cycle, this one, where it has to go to the retinal pigment epithelium, this takes hours, whereas your recovery time is too quick. All right. So then transmembrane potential variation. Why should rhodopsin get phosphorylated? Many times it is observed and oligomerized. Why should many rhodopsin come together? So how can we explain why should transducin change, alpha transducin change its affinity from GDP to GTP upon light infringement? Now, so here, the Murburn model is like this. Light hits the retinal pigment in rhodopsin, makes superoxide. It can easily make that. So GDP gets phosphorylated. Transducin, that is inactive alpha, GDP becomes GTP, and then GTP becomes unbound. This serves as the messenger, not the alpha transducin itself detaching and going as a messenger. Protein complexation and all is too much. It doesn't make any sense in time frame. The protein molecules cannot go like that, and it's too deterministic. So this small GTP can go and go to the immediate beta transducin, and it gets hydrolyzed. Then that GDP releases. Then that goes and interacts with the phosphodiesterase, which is like a clipped molecule, which releases. This is it. So another version, you see, effective charge separation occurs. This is the basic Murburn theory. So from light, you have a direct oxygen-aided electrical transduction. That is something there was no charge. Now you get a charge. So this is mobile, and that will reflect in a charge. So this can also phosphorylate. A GDP to GTP conversion is easy. Now you get GTP, and then it can give rise to the cascade. Now, how do we show it? Very clearly, what are the evidence supporting it? This, if conformation change and all that is so important, you would not expect this to be varying very highly, but then the rhodopsin structure is not highly conserved. All right. Oligomerization is a ploy, as we have seen in respiratory system. Many respiratory complexes come together to minimize the radical production zone. So one radical can be immediately scavenged by the other redox active moiety. And you can see the structure the reactive species can easily go. And that explains the proximity, the GDP, the docked GDP. So picomolar to GTP, the molecular affinity changes. So alpha transducin becomes, after phosphorylation of the bound GTP, it loses the affinity. So GTP detaches. So GTP detaches. You see, now you can explain the structure of, now you see here, alpha from here to the whole, it detaches immediately beta. Here currently there is no role whatsoever given to this. They don't know what beta GTP does. So the Murburn model clearly gives a very clear solution to that. That ROS comes here, I mean, the GTP detaches and their GTP is hydrolyzed. This has got very low affinity for GDP, but higher affinity for GTP. So docking very clearly gives the structural, the binding affinities, the signaling cascades, and now here also phosphodiesterase, same, all predictions of Murburn. I told Danny, check this, and everything came as per the prediction. All right, I predicted this would be like this could be like that, looking at the structure, just by imagining that oxygen is directly involved. So there you go. That's a very simple cascade for visual cycle. You don't need protein complexation, recycling through the retinal pigment epithelium. This is very fast. All right. So ROS can immediately cycle the electron deficiency and all that. So this is the same thing, same retinol. The carotenoids are present in photosynthetic pigments also. In that also, the same mechanism. All right. You don't need to invoke upon conformation change. So I think we are already close to lunchtime. So what I'll do is I'll go quickly over electrophysiology. I'm sorry. Neurophysiology and electrophysiology, we'll go quickly over. Now, classical theory is in homeostasis. The membrane protein somehow can quantify, recognize, and translocate. Then there are pumps, porters, and pores. And then somehow, with all these things together, a cell membrane works like a very intelligent system. All right. So the classical perceptions in electrophysiology is like this. There is a difference. There's a high amount of sodium outside, lower inside. So that gives an ion differential. And so in the, there is Jensku found this sodium potassium pump, which pumps sodium outside and potassium inside. That results in this kind of a status. And then calcium also found. So this pumping is the salient hallmark of cells. It uses a huge amount of ATP. All right. So this is the classical setup. And now when you have something like this, there are leak channels that push back the potassium out, and then that results in a negative potential of -70 millivolt, which is the resting membrane potential. Now, when there is activity, when the signal goes through the Hodgkin-Huxley membrane, neuronal model says there is sodium gates opening, coming in, then gates closing, and then potassium going out. And this goes on in a very orchestrated manner along the neuronal membrane, and this gives the action potential. This is the classical way in which your nerves are supposed to work. So this is the one that started everything. In 1950s, Hodgkin and Huxley got they did this work, and in 1960 or something, they got a Nobel. After that, 9 Nobels were given to justify further enforcing these kind of ideas. Membrane. This is the Cambridge School of Thought. So basically, transmembrane potential can arise in a wide variety of ways. And the difference in the ions, basically the ion movements, the cations are the rationales for the membrane potential difference in the classical theory. We cannot blame it also. They found that certain ions were where more mobile, supposed to see more, going across membranes. But then if you see basically the concentration difference and all that, somehow it doesn't make sense. And then there is what would happen if you understand ionic pump without the channels. It says it pumps out and then there are channels that bring it back. So somehow it doesn't look very, very appealing to me. Basically, this is the one that looks like a pump, kind of a pump. It's like you pump it. So first it binds sodium, 3 sodium inside, and then it uses ATP, pumping the sodium out. And then somehow it changes the affinity for that same binding sites. You get the same potassium coming back inside, and then things like that. So Freeman, Cope, Ian, Glenn, Tussell was apparently settled by Jensko's discovery. This guy, I think he committed suicide because of the issues, Freeman, Cope. So if mechanism of the sodium potassium pump, NKA is called Na potassium, that is K ATPase. That's what is abbreviated here. Is it a molecular motor? If it is a molecular motor, you would first need it to bind ATP. There should be gating, some gate should open, it should take 3 Na in, then that gate should close, then it should go inward, then some gate should open, it should throw it out. All this, it should prevent. It should be a very, very, very sequential, one after the other, very clearly designed kind of hyper intelligent kind of a protein. So, but the point is, when you see the structure, you don't see anything that resembles that requirement. This is what it is. This is the NK. It's a very porous. I have multiple pictures to show. But then let's just imagine, what if, just think about it from a layman's point of view. It says 3 sodium is pumped out, 2 potassium comes in. And if it has to maintain a steady state, that means the active role should be countered by a passive mechanism. Only then you will maintain that. So it's just a mirage going around. There is no evidence. It says passively it will be coming back. And is there an evidence? Is there any evidence to show that 3 sodium is pumped out, 2 potassium is pumped in? Nothing. It's just a consensus. And also, it doesn't make any sense in terms of mirage. You remember the picture that I showed you in Escher, the water flow. It just means this: 1 potassium is less, 3 sodium goes out, comes less. So there is some kind of positive outside. An image is created. The active role must be countered by the passive role. How does it happen? Why should it happen? It just happens. So it's a wasteful process that just, have you heard the Sendil Countermany jokes? They have banana in one hand. He goes and gives the banana. How much for this? 30 rupees. He says, you give that. And then how much for that orange? 30 rupees. You take that orange, 2 oranges. Then say, how much for that apple? You take that apple and then give back the orange. Where is the money for the apple? Somebody asks. Say, I didn't take, I gave 2 oranges in lieu. And then when you ask for the orange, you say, I didn't take the orange. Some kind of a vague accounting. This is precisely that. So the problem nobody looked at is the thermodynamics of the ions involved. Very clearly, there is very clear difference. Potassium is the larger ion. So sodium ion is the smaller ion. These are the different features and the solubilities in terms of the molar. You see, while chloride ion has comparable values, phosphate ion, acetate, carbonate, bicarbonate, carboxylates of proteins, all of them preferably solvate better with potassium. And potassium has the delta G of formation of this potassium ion is this, much different from sodium. So these aspects and its interaction with the reducing environment and the interaction with oxygen is what leads to potassium's favorable presence in the cell. It's not other. And then the the transmembrane potential is by virtue of the small amount of ROS. So we have gotten a simple equation based on that. And this was this scenario before Murburn concept came into it. There were three theories. There was the membrane pump theory, the association induction hypothesis, which was formulated by Gilbert Ling, to which Laurent Jakin, Hirohisa Tamagawa, whose message will be played here. He is also a very passionate person. He has got an equation that explains this part. That is how transmembrane potential is made. There's a GHK equation, Goldman-Hodgkin Katz equation, which defines the resting membrane potential. So Tamagawa had made a Tamagawa equation based on this theory. So along comes, see here ions are pumped, here ions get absorbed. In this, there is effective charge separation, and this also accepts adsorption. It doesn't say that adsorption is not an important thing. So what we showed, Hirohisa Tamagawa in Gifu University showed, based on a simple suggestion, he showed, just reduction of ROS can give transmembrane potential fluctuation. So the reality is that across spectra, the ionic differentials don't really correlate with transmembrane potential. What the classical theory does is it creates a membrane potential constants and somehow fits experimental data. So I'm not going into the details of this. This is the structure of the pump. You see how porous it is. How porous it is. How can there be any gating in this? It's just people seeing things, emperor's clothes, it's a very fine fabric. You will see only if you're a connoisseur. That kind of a thing. I'm a young kid who says Raja Nangai. So here are the cardiotonic, toxins and all that that's supposed to bind. Here are the channels, potassium channels. Channels are not made through the protein. They are made between the proteins. So here is where Bob McKinnon got the Nobel, where potassium is somehow stabilized in certain ways, in certain dimensions. The only problem is if this is selective for potassium, the way McKinnon says, I made calculations. It will take at least hours to years for one potassium to go through if it has to be selective in the way it is. Binding requires time, and selection, all that requires time. It has thermodynamic cost also. Dehydration of ions, then rehydration of ions. It's not that easy as people are given the. So what the Murburn view says is this one. The phospholipid, there is electrostatics, the phosphate ions of the lipids, they keep the anionic part inside. And so you have greater propensity for the potassium to be inside more. All right, so here is the structure. There was, once again, there was this is the main alpha protein subunit. There's a beta subunit. There was no role attributed to it. We saw that it has a high amount of lysine and arginine. So it's a ROS recruiting system which can sieve, thermodynamically sieve sodium and potassium. That's what it does. See, this is a very simple mechanism how some proteins, amino acid side chains, positively charged amino acid chains, a preponderance at a very simple location can achieve such kind of sieving. So there is a large amount of data which shows that phosphorylation on NK is also seen at the intermembrane, transmembrane region. That is how we have ROS here. That ROS recruitment simultaneously leads to this also. So like what Danny showed in LDH, this NK modulator, such diverse range of NK modulators. See the molecules that are supposed to modulate. They are all redox active. That's the only thing. It's not a binding based one. So simple NK structure, oxygen sensitivity. It's an oxygen sensitive enzyme. Uncouplers, those uncouplers that were supposed to work in all the other systems also work here. And NK can also do ATP synthesis and transdermal peptide. This peptide has lysine, histine. This is also active. It also messes with the activity. So application of Murburn, some people with a neural disorder, you can lithium, its size is quite different. The size is here, but the redox potential, you see, it's closer to the one. Where is it? This is sodium. This is oxygen. These are the values. Lithium, the value of the thermodynamic delta G value explains closer to the value of potassium. So you have better explanation with that. See here, this is the cardiotonic steroids that digoxin, urbane, bufalin, toad, those toxins. Now they are supposed to go and bind at this place and somehow block those specific gates and all that. No, this is, in that case, you should have steroids also messing with it, because it's the basic fabric is that of a steroid. So we, there are molecules that don't have the steroid backbone, but they have the that what is that called? Quinone, flavone. What is that? What is this molecular structure? Cardinolide and bifidinolide. This structure, this is reactive oxygen species, or DRS, interfacial active. So any molecule that has that, it serves. So many molecules that have. So it can both position better there and also do interfacial activation. So in Murburn model, NKA is a interfacial Ross dynamic modulator. I'll quickly go over neurophysiology. I'm sorry. I've already done this. This is what it is. So, but the problem is, what are the critical question that you need to ask? The simple, this is called Fox microspheres. Those that don't have pores, all those, they also show exactly the same kind of action potential without pumps and channels. Why do ROS affect these outcomes? These are the questions that need to be asked. So we recently modeled based on Murburn. We modeled reaction, relaxation, transport based, electronic, not ion pump based, not ion movement based mechanism. This is the equation. I'll not go into the details. So basically what happens there here is, in the classical primary carrier is ions, in its electrons and DRS. Voltage is the driver here. It is a derived one here. Energy source is ion gradients. See, it's cyclic. Ion gradients give voltage. Voltage gives ion gradient. There is no causation, no cause and consequence correlation. Continuous redox metabolism. Oxygen's role is direct here. Then the ion flux is a primary. It's a secondary buffering activity. Prediction, it requires channel gating, just redox sensitive excitability. So take-home messages. Sorry, there was another picture. I don't know. I made this yesterday night. So something has gone. There was a picture to show you the basic outlay. But then the classical model, ion channel centered explanation. In the Murburn model, it is neuronal electricality emerges from redox mediated electron dynamics. Links metabolism, oxygen, reactive species, and excitability within one framework, makes experimentally testable predictions on oxygen, ROS, ionic strength, D2O, and temperature. Lots of data we have. This equation is beautiful in the sense that with 4 or 5 variables and parameters, we can catch the whole transmission velocity effect of all these parameters, whereas Hodgkin-Huxley requires 40 odd terms to get there. And there it is, ionic currents. Just imagine this. Now you have suppose you imagine this is the axon. Hodgkin-Huxley is about this movement of ion from here to there. Murburn is from here to there. Where does the message go? Goes from here to there. Very simple. So while it measures ionic current based on transmembrane voltage fluctuations, this is a oxygen-centric electron-centric process. I'm sure you will have a lot of questions. I open up. That's it. This was put together because there was somebody else, somebody else. I had to somehow fuse 3 agenda. That is vision, electrophysiology, neurophysiology, all just in one night's preparation. So that's why I just put certain things that are clearly discernible. I did not go too much against the neuronal model because it's very touchy to some people. But then let's see the point that to have ions go in and out, first you pump ions, generate a voltage. Then that voltage controls voltage gated channels. I mean, on the same membrane. How does it make sense? It doesn't make sense to me, at least. So that's why I questioned it. So there is an active process which generates something, and the passive process exactly counters that in various conformations, in various geometries. How? Why? Just because some people said. So here you have any signal, whether touch, vision, everything changes oxygen superoxide equilibrium. That's the most fundamental way. The only thing you have, every cell, what comes in? Oxygen and nutrient. And what goes out? Carbon dioxide and water. This is the machining logic of every cell, whether it is a neuron, any cell. That's how it works. So it has to maintain. So changing the potential, the electrical activity is an outcome. It is not the driving force. Suppose if a scooter runs, imagine a scooter runs. It makes sound. It gives out smoke. You don't say when it makes a huge noise, you don't say, oh, the scooter runs because it makes a huge noise. It's just an outcome. Similarly, the voltage that you get is a dissipative outcome. It's such an associated phenomenon. What really is driving the process is the redox process that occurs within the cell. The complications are too much and too little time for me to present at this juncture. I finished my talk. We are all waiting for lunch. I know 1:45 already, late already. I can take a few questions. Sir had posed me some interesting questions. Krishnakumar sir, why at the nodes of Ranvier, he works in this area. Why are the channels very highly clustered there and all that? Those kinds of questions. We have to spend a lot of time. I have not really thought about it. But what I have done is address the basic issues in the sense of cycling, working, speed, composition, neuroglobin, the classical theory. Like in the muscles, there is myoglobin. In blood, there is hemoglobin. There is neuroglobin. The classical theory has nothing, no role. So here in the Murburn, they work as pit stops in this relaxation transport equation that I showed there. So it somehow appears to be much better than the classical one. Yes, there is online. Can you show who is online? Mahendra Kavdia teaches this in Wayne State University. Is Mahendra online? He's a co-author. Mahendra, can you say? And Taufia also. Taufia, Mahendra can start. He's a professor in Wayne State University. He is a collaborator of mine. We have done this work together. So Mahendra, would you like to say something, please?
I would just say, like, Murli, you have some God-given gifts, and I think you're doing really good, and you'll change certain things. Only thing, because I teach and these things in electrophysiology, and I have really understand the sodium potassium channels and electrical currents and all those things. So what I can tell you is my collaboration with you is mainly to make sure your numbers are correct and your basic things are correct, and to now I haven't found any faults or something, so which is good.
That's good. Thank you. Yeah. Taufia did her master's in VIT, where I used to teach. Now she went to Rostock, where she has been doing this electrophysiology, neurophysiology work. She works with computational simulations. Recently, the equations that we derived, she did some simulations with that. We sent this paper to PNAS now. Our papers, both papers came back after about 4 weeks. Standard rejection. No comments, nothing whatsoever. Today, I got my second paper of the flagellar model. Also, I sent 4 papers, 2 on bacterial flagellar model, quantitatively sound, beautifully derived. Both were rejected without any rationale. So Taufia worked on that. Maybe Taufia, you want to share some comments?
First of all, I would like to thank Molly sir for giving this opportunity to be a part of this interdisciplinary work. And it was something which interesting to work on, because I have been brought up with the old classic neurophysiology concept. And when he introduced me to this new, it took me some time to actually go through it and try to accept what he wants to say. But eventually he was succeeded in actually convincing me. And I'm really glad that I am a part of this whole framework. And one thing which I wanted to actually tell, I think it's a right platform, because it can be used a lot in some sort of neurodegenerative diseases like ALS, and I am trying to simulate that as well. And to some extent, I have got some positive results, and I'm just trying to. I can share this with Melissa later if he is interested. But I think it has a lot more. The scope is really vast, I would say. Because we are moving into really a paradigm shift in this area, and I know it's not easy for most of the people to accept at a first go, but I think it has a convincing approach, and eventually I think people will definitely accept it. So I'm really thankful to Murli sir to give me a chance to be a part of it. Thank you so much, sir.
It's wonderful to see you, Taufia. It's how many years? 2008 to 2010, right?
Yes, 2008 to 10. Yeah.
Almost 16, 17 years. Yeah, I'm so happy. It's a teacher's joy when your students who are, they come around and accept your ideas. And that's how I said that, earlier, tigers, I said this yesterday, tigers are much stronger. How do we pass on the baton? Our kids, they take up, they spread the news. And wonderful. I'm happy that you took it up and you have done that ALS work, and hopefully this mathematical thing that we have together, and also the fundamental ideas, textbook ideas, will give us greater insight into the neurodegenerative and all those disorders. So thank you. Mahendra, you want to say something else?
In terms of I have been working in nitric oxide, oxygen transport, and reactive oxygen species for last 25, 30 years. And it has always been a question why reactive oxygen species has to be there, and what is the role of these. And more recently, people are started thinking reactive oxygen species as a signaling molecules, and your Murburn concept comes completely in terms of that perspective, that reactive oxygen species are very useful for human physiology.
Thank you. I completely forgot. Sukumar sir is my co-author in this neuron paper. I'm so muddled. Sir, Sukumar sir, can you hear me? Sir, can you come on?
I can hear you. I can hear you.
He is the guy who fixed my errors in this first. Sir, please, can you come online? Can I see her? Yes. He is a quantum physics chemist. So earlier he was also looking at me with a lot of askance. Somehow I convinced him to join.
Well, I still look askance.
That's what I do. Yeah.
I wouldn't call myself a convert yet.
Yes.
Although I did enjoy many of the talks today, and things started making a lot more sense to me. But as you said, I'm a quantum chemist. I'm not a biologist, not even a biochemist. So many of the terms, many of the ways of thinking are new to me. My role has been essentially to play the devil's advocate, to try to poke holes and things like that. And I think it was it might have helped in some way, because as holes
It
get
has.
punched,
You bet it has. You bet it has. And,
but still there are things that need to be patched, like where the equation comes from. I mean, you started out with a derivation, but that's not really the thing. And when Abhijit introduced the nonlinear terms,
that is the last part.
But we have to show where that comes from, from a fundamental level, right? Right now, it's a phenomenological equation.
Yes. But it's anyway better than what we have. Yeah, give it, sir.
Yeah, I'm not the person to speak about.
Okay. Fine.
You have to invite one of the defenders of Hodgkin-Huxley to come and this thing, and then you can poke holes. I'm nobody.
Yes. After the two talks by Abhinav and Danny, he sends me a message. Your kids do a lot better than you do. Himachal Pradesh now. He was in US for a long time in how do you pronounce it? Rensselaer, yeah. He was there. So yes, thank you. Please stay on.
You are too passionate. In some ways,
I'm
it's good.
Passionate in whatever.
In some ways, it's good. In some ways, it kind of starts to trivialize the message when you're dealing with a professional audience. With students, to inspire them, these little analogies, help. But if you try to put that in a paper like you did, but anyway, we can take that off.
Wonderful. So Professor Yanyu Wu is actually a photosynthesis person. It's the next session. Professor Wu, are you there? No, I think he might.
Yes, I'm here.
Yes. Wonderful. Can you show us your? I want to see you if you can come. We have
sound is not clear for me. So can you increase the quality of sound, please?
Volume.
We can hear you all right.
I thought you could hear
we
me
can't see you
without the mic. I speak so loud usually. Please, from Chinese Academy of Science. Please.
Yes. Hello.
Yes, we can hear you. We can hear you. Hello.
I think he's muted.
Yes, to unmute.
Professor Wu, you are muted. Can you unmute your mic, please? Anything else you want to say, Taufia?
Hello.
Yes. Sorry, Taufia.
Yes.
He's come back. Yes.
Sorry, since it's first time to use Zooming, we usually use Tencent. So, hello.
Yes, we can hear you.
I have a problem in connections, I think.
Professor Wu,
can you please give me some opportunity to reset my connection, as I have a problem in connection now?
Okay. Maybe we can reconnect after some time in the photosynthesis section. Is that okay? After some time, he's a plant physiologist, and that's the next session. He has joined earlier. He is from Chinese Academy of Sciences, and we are one of the few who had some contesting issues with the classical model of Z-scheme and some bicarbonate fixing mechanism, bicarbonate utilizing plants. I think Professor Wu can connect later. He has some issues. So Taufia, if you want to say something, you can say, and we'll come back to the place.
I would like to ask Melissa whether it's okay if I present this concept here in the university in the neuroscience department.
Absolutely, my child. Go ahead.
Yeah, thank you so much, because I want to with the ALS, I want to give them, because it has a big a neurodegenerative faculty here. So I want, and it's an upcoming seminar. So I thought that why not to introduce this concept here.
Go.
Thank you so much, sir.
Yeah, I called her my child, but she's not so young.
So any questions, students?
Good.
Students, yes. Any questions?
Danny, you're raising your hand? Oh. Lunch. So, sir, please.
So it was all a wonderful presentation, and we had a great discussion. I think the numbers sir has left. I think the younger generation has to take it further, and particularly in the neuro field, because central nervous system is one area where ROS is produced in huge quantity. Retina is part of the central nervous system, so it all explains very well. And secondly, you have to understand that when you think to move your feet, this happens in less than a second, right? The moment you think you can move your feet. Now you think about that, if it is the ions which are moving across, if the ions are the one which is moving across, it will take more than seconds to reach the signal to move your muscle to make a movement, right? So then how this thing is happening? Think about it. If it is ions, in a potassium ion, sodium ions, etc., is moving across, the sodium and the potassium channels, you have the nodes of Ranvier, which are, I call it as amplifiers of the signal. And if they are sitting in those areas, if it is movement of the ions across, it is practically very difficult for you when you think momentously you can move your hand. The best example is that all of you have watched cricket. The guy who is standing right in front of the batsman, how he does the reflex action catching a ball which is coming at more than 100 miles per kilometer per hour, hit, and then momentously catching the whole thing. If the ions have to move and then do the whole job, it is practically difficult. So then you have to have an explanation for this, because the thing is happening. So what is the other explanation? Here comes the electronic movement. And this is what needs to be explained in detail with a much more experimental evidence. And in neurodegenerative disease, because that's one area which we work heavily, we can see all these channels. It's beautiful clusters. Sodium channels are clustered beautifully at the node. You can see in the juxtaparanode, the potassium channels in between the Casper protein, etc. They're all organized beautifully. But the moment demyelination happens or the neurodegeneration happens, all these things are declustered completely. And you can see the no impulse conduction, etc. So what these channels are doing, actually, they are providing as a platform for the reactive oxygen species to play their job. This is what we think, and probably you may agree with that. Thank you. So, I think if nobody has any questions, anybody has any questions? If nobody has any question, we will wind up. And thanks once again, primarily for inviting me. Thank you very much.
There's a break now. Taufia, thank you. Thank you, Mahendra. There's a break now.
Okay. Good luck, Murli.
Thank you, Mahendra.
Hello? Hello? Hello?
Professor Wu, hello.
Hello,
Professor
I'm so sorry for interrupting the connection. As you know, we in China using a VPN,
No
so
problem, no
VPN is
problem.
not VPN is not stable at this moment. So we are apologize for this
No
technical
problem.
problem.
No,
Yes.
Professor Wu,
By the way, my name is Mo. I am one of the Professor Wu student.
okay.
As you know, he told me just I can participate as I have some good background in photosynthesis. So it will be very good thing to join for you, professors.
Yes. What's your name?
My name is Mo.
Come again?
M-O.
Mo.
M-O,
Okay.
just Mo.
Yeah, Mo.
Mo, yes.
Dear Mo, we have just broken for about 40 minutes for lunch. It's 2:00 here.
It's okay, bro.
We will start at 2:45 in about 40 minutes. Would you be joining then?
Yes, I'm be available. I'm just totally
Yeah.
available for this
We will
meeting,
join
Professor.
after 40 minutes for the next session on photosynthesis. Okay?
So 40 minutes from now.
Yes, 40 minutes from now.
Okay, I will be there. Thank you very much,
Thank you.
Professor.
Thank you.
Very much. Thank you.
our correspondent. So anyway, since the session is going to start, I request the chairman, Dr. A. Lada. She is the Director of Research of the Kerala Agricultural University, to occupy the chair. And I also invite Dr. Anitha Karun, former Director, ICAR Central Plantation Crops Research Institute, also to join her. And I also call Vasudevan sir, I think. Vasudevan. Where to go? Where to go? Sir, please come. Here. Okay. Then also Murli ji. You will be speaking, sir.
Hello. Hello. Can you hear me? Good afternoon. We start our session now. Sir said 2:40. We are starting around 2:50. So this is the last session. Some kids have already left. It is quite understandable. So now we start off with two messages. One from Professor Hirohisa Tamagawa. He's basically a mechanical engineer who works with polymers. And he also has another dimension. He works on the electro and neurophysiology. So he has been collaborating with me for the last 5 years. He would like to share some message. He was supposed to come online, but his mother met with an accident. So, yes, please, Professor Hirohisa Tamagawa.
Hello everyone, my name is Hirohisa Tamagawa. I am a professor at Gifu University in Japan. Although I was invited to this distinguished meeting, I have unavoidable obligations until August 9th, as well as my personal matters that require my attention. Therefore, I regret that I am unable to attend this meeting. I sincerely apologize for my absence. I would like to briefly show my impression to the Murburn concept. The Murburn concept is an entirely new physiological theory because it represents a fundamentally different way of thinking about physiology. Many researchers are understandably hesitant to accept it. However, I believe that many scientists have already recognized the limitation of existing physiological theories. At the same time, I understand that it is not easy to move beyond the long-established ideas of physiology and embrace new frameworks such as the Murburn concept. I sincerely hope that the Murburn concept will open the door to a new era in physiology. Yeah. Thank you. Bye-bye.
So, Markov, here is PowerPoint, please. Next is Dr. Marco Fiore. He is an Italian researcher from CNRS. So he has a PowerPoint. He has sent in a PowerPoint. He has gone away with his family. 2 people have gone away for a holiday in Alps. So that's why they are not able to join. So here he is. Yeah. Please.
Good morning to everybody. This is Marco Fiore, speaking from Italy and coming from the Institute of Biochemistry and Cell Biology of Rome in Italy.
Yeah.
We are going to speak about the Murburn concept. The question is, could the Murburn concept reframe mitochondrial oxidative processes to frame Murburn concept as an intriguing oxygen-centered hypothesis? We are going to interpret as that mitochondria as controlled oxidative spaces Mitochondria are not just ATP factories, they are structured oxidative microenvironments where electron flow, oxygen reduction, ROS generation, proton gradients, and the membrane architecture coexist. If ROS are not present in mitochondrial respiration, are they merely accidental byproducts or are they part of the operating logic of the organelle? And now, what is the Murburn concept? As far as I know, the Murburn concept proposed that diffusible reactive species are not just toxic leakage products, but functional participants in redox catalysis. The question is, could mitochondrial membranes act not only as proton barriers, but also as radical reaction boundaries that confine and regulate oxidative chemistry? Indeed, the Murburn refers to mild container redox chemistry involved with diffusible reactive species. Murburn proposes that ROS, RNS, and other diffusible radicals can mediate catalytic processes outside the classical enzyme active site model. In mitochondrial oxidative phosphorylation, the Murburn model proposes an oxygen-centered radical-mediated explanation rather than a purely proton-centric chemiosmotic one. This should be considered as an alternative and debated framework, not the standard consensus model. We are approaching the core of this business. Murburn and mitochondrial membrane stability. The key question is whether or not controlled ROS chemistry could help maintain membrane function, while uncontrolled ROS causes damage. In oxidative stress, controlled ROS may support redox signaling and metabolic adaptation. Excessive ROS can damage proteins, lipids, and membranes. The Murburn view would interpret mitochondrial ROS as potentially functionally when spatially and kinetically controlled. Intermembrane composition, crystal geometry, and respiratory complex organization may determine whether or not ROS are useful signal or destructive agents. In the Murburn concept, what determines the switch between beneficial ROS-mediated regulation and destructive oxidative membrane damage? Crystal corpuscles and selected oxidation. Crystals are indeed good structure for asking whether or not subtle radiochemistry shapes mitochondrial function. Crystals are specialized inner mitochondrial membrane domains containing respiratory complexes and ATP synthase. Crystal junctions can restrict diffusion and create local electrochemical differences. ATP synthase dimers help shape crystal curvature and organization. Murburn raises the possibility that crystals also help localize radical-mediated oxidative reaction. I do have so many questions. For example, could the mitochondrial crystal be understood as organized Muir zones where membrane architecture controls the balance between energy production, redox signaling, and oxidative damage? In conclusion for this very short presentation, the Murburn concept does not simply ask whether or not mitochondria produce ROS. It asks whether or not controlled radical chemistry is part of how mitochondria work and act. The critical experimental challenge is to distinguish regulated oxidative microchemistry from accidental oxidative stress. And thank you so much for your patience.
I think with your permission, we can move on to Abhinav. Would you like to start? Huh? Slides are not ready. All right. Yeah. All right. You want me to talk?
Hello.
Hello.
Um, uh, I just have a question for Professor Marco. Can you allow to me?
Yeah, if you want. We are going to talk about photosynthesis now. Is it okay if you can to that, or you want to ask before that?
It's okay. I just I can wait, no problem. So it's a simple question. So I can wait for the photosynthesis lecture.
Sure.
Thank you very much,
Thank you.
Professor.
Yes, thank you. Thank you. Thank you. So then give me the slides. I can do the whole thing. Have you sent that? No? Okay. In that case, you give it to him after you complete. After you complete, I'll start with my talk. Hello. Yes. Everything is done. Good afternoon. Now, perhaps this is the last session. There are multiple aspects that we'll cover in this. We'll do the most important reaction on planet Earth, which Indian Council for Agricultural Research stands fundamentally. Of course, here it is marine fishery research. Photosynthesis. All right. It gives us the food that we eat, the oxygen that we breathe, primary source of carbon-centered life that we see on planet Earth. So, most of you know from your school or higher secondary education, you have the chloroplast is a place where photosynthesis occurs, like mitochondria is the place where respiration occurs. So there are grana, there are stroma structures like this, stacks of lipid-based membranes on which photosynthetic proteins are spread. Now there is a lot of information available where you know which what kind of proteins are available more, where, which part of this grana, which are greater distribution densities where. So this is the ultimate fixer of carbon in the light reaction, ATP. This is what my concern. I'm not involved in Calvin cycle. We are dealing with how NADPH, the reducing equivalents, and ATP, the energy currency, and oxygen are produced. Photosynthesis, the day reaction, 3 fundamental products: the reducing equivalents, the phosphorylative energy, and the molecule of oxygen. This is produced from water. Currently, the mechanism is highly deterministic. We'll go. All of it starts from the experiment of Emerson, a very famous experiment, Emerson experiment. What was done was oxygen evolution was seen. There were, at that time, it was known that there were 2 types of photosystems. One that primarily gets excited at 680 nanometer, another at 700 nanometers. So they first excited at 680 nanometers, they got some amount of oxygen. Then they could excite it at 700 nanometers, some amount of oxygen was derived. The interesting thing was when the system was excited at both 680 plus 700, the amount of oxygen that you would get, it was more than the combined of these 2. So what those scientists did is they said this is the stoichiometry, that is the ratio of the overall reaction, wherein first water was broken down at photosynthetic system 2, PS2, 680 nanometer, and then the electrons were relayed serially through some intermediary proteins and small molecules like plastoquinone and plastocyanin. This is a protein, this is a membrane embedded quinone. So then finally coming at P700, that is photosystem 1, to reduce NADP to NADPH. So this is a serial electrons transport chain. It was called Z scheme. Why Z? Because goes like this. So then the ratio was 2 molecules of NADP plus held at reductase, 4 electrons from 2 molecules of water held at PS2 to release 1 molecule of oxygen. That is 2 molecules of water were held here to release 1 molecule of oxygen. Now, the problem is, this is something that violates basic awareness of current flow, or basically traffic. You imagine there is a reservoir of electrons, photosystem 2, photosystem 1. If you connect them in series, what will happen to the electron flow? Basic 10th class understanding of electronic circuits. When you connect 2 resistances in series, what happens to the overall resistance? It increases or decreases? Let's have some interaction. It's already afternoon. Resistance increases, so current flow will decrease. Is this a good idea to connect them in series? So now suppose if I had to get greater traffic from here to say, madam, would I make one long road or multiple parallel roads? If I wanted to send more traffic, I would have multiple parallel roads. So it was a very wrong idea in terms of either fluid flow, traffic consideration, or even electronics or fundamental electrical connections. It is not a good idea to connect in series. It's completely wrong. So to explain Emerson experiment, they connected photosystem 1 and 2 in series. Nobody on this planet Earth can ratify this rationale. Govindji, who wrote the textbook on photosynthesis, his responses are officially noted on my YouTube website. All right. So this is the general idea. This is how it looks like. Proton pumping. So the whole point is when chloroplast size excluded volume, there's hardly any protons. So how will it pump out? And the electronic circuitry goes down, up, out, all over the place. There's no directionality. How? So cyanobacteria can both respire and photosynthesize. This argument I had said earlier. So, and I also talked about the commonality of certain agents. How will it know what to participate in? All right. And we also know that a membrane cannot just pump ions both directionally, different kinds. There is hardly any logic. So the idea that I wanted to very clearly say is whatever is given in textbook, you check out. See, this is 4 photons here, 680 nanometers, 4 photons here, 700 nanometers. So that's supposed to give this. You put that equation and you check it out. It requires 1464 kilojoules per mole. 4 photons each of 680 nanometers and 700 nanometers only give 1387 kilojoules per mole. So I don't know what they did to get the, even if you accept proton pumping as a energy conserving gradient forming mechanism, this reaction is unviable even with the basic treatment. Then deterministic key carriers. If you see the overall system, there are multiple. This is a quinone. This is a membrane quinone. I had shown its structure yesterday. Plastoquinone is a little different from coenzyme Q, but they're still quinones. Large lipids that span the membrane. It won't move like that. It has no way to deterministically move. And then this is a soluble molecule. How do they move like that with an errand? And particularly now that we know the distribution, photosystem 2 is deep inside the grana, photosystem 1 is outside, cytochrome B6F is outside. So the plastoquinone should jump thylakoids. How does it happen? All right, so how does the proton pumped here comes to the close complex Y that is here? It doesn't, this kind of setups, the architecture doesn't agree. And the very important thing is plastocyanin, its topology. Danny explored this. He shows that, you see, if the electron transfer, if plastocyanin takes the electron from photosystem cytochrome B6F to photosystem 1, like cytochrome C takes from complex 3 to complex 4, similar parallel here. You would imagine that it would be conserved. Its topology would be conserved if the binding based, there is no conservation. And then plastocyanin is available at very low concentration, 1 micromolar. If it is so important, you should have it at higher concentration. Most importantly, they made in Arabidopsis thaliana, they knocked out plastocyanin completely, complete knockout, no plastocyanin, yet it works beautifully. So the plastocyanin or cytochrome C here, this is cyanobacteria. plastocyanin. This part was knocked out. Sorry, I shouldn't have shown the respiratory chain, plastocyanin here in plants. Then, if you see the structures of photosystems and the light harvesting complexes, the colored pigments are scattered all over the place. And there is hardly any covalent tagging or tethering. So they just occupy random positions, it looks like. There are even colored carotenoids, even in cytochrome B6F, which doesn't have any, photoactive responsibility. So why or how? And then the action spectrum also, there are, doesn't really, there are so many, there is decent amount of photosynthesis, not that efficient, even in non-PS1, PS2 regions. So then here comes the Murburn model. Rather than proton pumping and all that, here what we say is light hits chlorophylls and carotenoids and xanthophylls and all those pigments and all those, they can excite and liberate electrons, which can be collected by NADPH and oxygen. So the most important thing is this is a highly decentralized parallel system compared to the serial mechanism and sequential serially ordered. Here, everything works independently. There is no necessity for that, what this scheme advocates. So here, cytochrome B6F, ADP binding, all those membrane extra subtensions, Abhinav showed that they bind ADP. So what happens is here, reactive species are formed. Reactive species can attack phosphate, or the electrons can go into NADPH. And when it goes to phosphate attacking, it forms ATP. It's a random delocalized thing. But then somebody would ask, why photosystems? They are effective charge separating agents, quite unlike light harvesting complexes. So let's see that's the one view. Here, I would say there are two kinds of reactive diffusible species produced. One that is required for the engine proper, and another for lubrication. Like for light harvesting complexes, they produce reactive species which help function at high photosynthetic times. When electrons get displaced, there is electron holes, they replenish that, but they are not good at effective charge separation. So photosystem 1 and 2 are for that. So this is yet another view. Here is a more temporal kind of view. What happens is diverse pigments photoactivated, you get effective charge separation and electron emission, giving us diffusible reactive species. This diffusible reactive species can give electrons to photosystem 2 and 1 again, and diverse pigments, they give it back, and then they can also react among each other to form water and oxygen also. And photosystem 2 can better use H2O because it has got a manganese cluster, which can afford better charge conservation. It can deplete peroxide also better, thereby liberating oxygen from there. So here, diverse pigments can also electron replenish with this. So DRS is formed, it can replenish electron holes. For tertiary processes, membrane proteins can use electrons, generate DRS. They can give to NADP to form NADPH and also get phosphorylation. So NADPH synthesis, oxygenesis, phosphorylation, transmembrane potential generation, thermogenesis, homeostasis, all mediated through DRS in the Murburn model. So the beauty of this model is that it is thermodynamically, mechanistically, kinetically justified. You see how oxygen evolution, it is not centered only on photosystem 2. If you see, the experimental observation doesn't mandate that only complex, photosystem 1 can. See, Emerson's experiment clearly shows photosystem 1 is excited, it also produces oxygen. So that is agreeable to us, the Murburn model. So here, what it's saying, oxygen evolution is decentralized. All right. When the manganese complex, the OEC, so-called water splitting complex, that can effectively deplete peroxide and therefore tug the thermodynamic pull. It can drive the reaction. So that, because otherwise the 2 electron formed of H2O2 will linger and that will react with molecules and then it will not proceed further to oxygen formation. All right. So you can see in many ways you can get oxygen formation. So a product formation is not by one route. It's like you have rainfall on the mountain, the water falls through multiple routes to come and assemble at a lake in the valley. So something like that. There are multiple ways, multiple parallel ways in which oxygen can be formed. Similarly, NADPH can be formed, ATP can be formed in multiple ways. And all their energy terms are very simple, highly exergonic. You can have ATP synthesis associated with oxygen evolution. So here you can easily get much lesser energy required for if you use hydroxide ion. Here you don't need 2 water molecules to be bound at one time. No such fastidious requirements. So in vitro oxygen superoxide DRS equilibriums mediate ET, electron transfer. We have shown through our 1.5 decades research, not one experiment, not 2 experiments, multitudes of publications we have shown. And then we showed also ADP plus phosphate in the presence of superoxide gives ATP, which is inhibited by cyanide. This was shown by Tiskevitz and Rao. They in 1987, they took chloroplast, xanthine oxidase and xanthine. They got higher ATP, which is inhibited by SOD. So clearly all the experiments that need to justify the basic foundation of phosphorylation in the Murburn model in the chloroplast is available. Now let's see the structure of proteins, the thermodynamic, kinetic. See, these molecules are all fundamentally bimolecular reaction, though I have written it like that. First this attacks this and then something is formed, and then that attacks another one and this is formed, and then the 2 radicals in that react together to form the third one. So they are all fundamentally bimolecular reactions whose reactions are facilitated by these kind of structures. This is photosystem 2, then this is NADH, NAPH dehydrogenase. This is photosystem 1. So you see, like I said, random scattering of chlorophylls here and there. You see the porous structures. If there are really beautiful electron transporting route, you will not expect such structures. And this is cytochrome B6F. It has carotenes here and there. There is no rationale why you should have subtending pendulous parts like this. Abhinav showed that they are, see here, ADP bind sites. Same case with NADH dehydrogenase, NADPH dehydrogenase. So all of this, the random distribution, the subtending, outside membrane subtending, redox centers, all that agree with the Murburn model. So they are low affinity binding sites. Pigments and Ross channels are noted in proteins. The latter is inexplicable if the deterministic electron circuitry were physiologically active. Very importantly, through 30 years of research, many people have shown that very diverse types of electron donors can function for both PS1 and PS2, photosystem 1 and photosystem. See, all these have been demonstrated to serve as electron donors. Electron donors and acceptors for PS1 and 2. Then E acceptors from PS1 and 2. So there is no real specificity for anyone to argue. Very clearly, there is no specificity whatsoever. And also, we had published with Professor Wu this fundamental major problem in photosynthetic chemistry. It was seen that bicarbonate, when you give to this system, it enhances the photosynthetic rate and oxygen release. So we showed that through the calculations that yes, it is somewhere between the values of energetic yields between the water, the amount of requirement for water or for hydroxide ion. So it can definitely, in Murburn, it doesn't require specific binding. So this can also work. So diversity of E donors and acceptors from PS1 and PS2 and all that discredit the Z scheme. You don't need affinity bound moieties to give. Bicarbonate known to enhance oxygenesis and Emerson effect supports decentralized oxygenesis. These are the two fundamental experiments that were misinterpreted for and which gave a misdirection to bioenergetic research. First is Jagendorf research. This somebody talked about yesterday. First it was done in chloroplast. What they did is first they took pH 4, they equilibrated there, and then transferred to pH 8. In absence, they just added ADP PI, and you could get ATP. So that means that a pH gradient works in chloroplast. Now, if you see the Murburn equations for that, it will tell you why. You see, plus this gives NADPH. Everywhere where it requires H plus on the left side, you give, it drives the reaction forward. All right. Even in the other one, ADP plus PI plus H plus, that phosphorylation also goes. Even this phosphorylation. In all this reaction, H plus can be a reactant. So if you give H plus from external source in a proton limited area, then you will get reaction. So that was a reaction experiment which had zero relevance to physiology. Zero relevance. Absolutely zero relevance. Then the another experiment that was done was called Racker's experiment. All right. Racker's experiment, what they did is, Ephraim Racker, he constituted vesicles and he added these different systems. That is, in one, he put only bacterial rhodopsin. In the other, he put only ATP synthase. In one, he put both rhodopsin and ATP synthase. And in the other one, he put rhodopsin, ATP synthase, and an uncoupling agent. Now, what was the observation? They said no ATP generated in bacterial rhodopsin, no ATP generated in ATP synthase, but ATP was pretty decently generated in rhodopsin plus complex 5, that is ATP synthase, but ATP synthase was, ATP was not generated with uncoupler. So they argued that uncoupler basically dissipated the proton gradient. Now, here is the biggest problem. Without complex 5, this gave 20% of the ATP that was generated in this system. So they conveniently oversighted that. Because they said complex 5 with this thing, if this gave 100 molecules of ATP, this gave 20. And this also gave some, but it gave lower than this. So disregarding observations, superposing one's own whims and fancies, Does not. This reaction, if you say complex 5 is the ATP synthase, why am I not getting in this? They say that this works as a proton pump. Proton pump creates a gradient and then that comes back. Why? It's a vesicle. So the point is, here you could see ATP synthesis without ATP synthase, so-called ATP synthase. That is the key observation. So they said this pumps proton. It's a very well-known thing that rhodopsin's Gibbs base pK is around 13. There is no way this can function. It is 5 pH units above physiological pH of around 8 or 7, 6. It will not function as a proton pump. It cannot function. So these experiments had zero relevance to physiology or they were misinterpreted. So this is the basic understanding. I'm not going into photosynthesis in greater detail. Fundamentally, we have shown that there is hardly any reason. Recently, Frontiers invited me to submit a paper and they just refused publication prospect. Outright, they rejected everything poor. I gave them a peace of mind. I said, we have already published this. And then when I told them like that, 2 new reviewers came in and they kind of said, this is a great work. So sometimes you have to put your foot down when you know you're confident. All right. So this is the photosynthetic work. The next slide, please. I have a lot of sorry. Sigmar sir doesn't like me, he was saying, yeah. So I wanted to just take a break and we'll get back to the talk. Now, if I say 4 elephants are found in Tibet, there are no elephants naturally found in Tibet. So now you have, imagine, sometimes when the elephants are found, sometimes a red Maruti is also found, Maruti 800. It has 4 doors, 4 seats. Now, sometimes also those elephants also have Kunyiraman, Appukuttan name tags in Malayalam. All right. So sometimes rain comes when the elephants are found there. So the elephants, they are noted here and within 2 hours, they are noted in Tibet. Now along comes somebody and they have a theory. The 4 elephants sit on the 4 seats. They drive down from Kerala all the way to Tibet in that Maruti sedan. Will you buy it? Will you buy it, kiddos? Will you buy it? Why? Why won't you? There is 4 seats. There are 4 doors, elephants, correlations. When the elephants are found, the red Maruti is found there. It has the Maruti has KL registration. KL registration. Why won't you buy it? You won't buy it because your ideas, day-to-day ideas of space, time, speed, abilities don't agree with it, right? Similarly, all these molecules like oxygen, they just can't bind and stay at one place. 2 molecules of water without releasing oxygen, they have to be brought together. Oxygen should not be going anywhere. All this is not possible. All right. These are all very superposition of one's own
whims and fancies on molecules. Cytochromes, plastoquinone takes an electron from one point, one photosystem, and jumps grana to go to some other protein. Why? Why should it? It need not. So when I presented this, if you are interested, you go to my YouTube, see what happens when I presented this in my talk with photosynthesis people. They start shouting, can't hear, because they have been habituated with that. But now you see how fast can a plastoquinone molecule go. Krishnakumar sir was saying ions cannot move that fast, as fast, this distance. For example, from here where your spine ends, the toe until the whole thing, there is one neuron. It's a complete cell. A signal comes from that to here through some ions and all that. Whole muscle contracts. How can you just calcium pumping and all that from here to here in fraction of a second? All these are just coincidences. Sometimes elephants were seen there, Maruti was seen there. There is no physical correlation between the size, the speed, the ability. Some correlations, that's all. And mere correlations are not enough to get sound thermodynamic, mechanistic, kinetic, probabilistic logic. Number correlation is not enough. All right? You need to make thermodynamic sense. So now I will go to the next part. What we do in this part, I'll just take what is the interesting or important part that tells you that this thing is wrong? One simple thing is enough to throw away this thing. Yes. Sorry. Online. Hello. Yeah. Sorry about that. You were right. Can you just take it? You are taking. He's taking. So, what was the most fundamental, elementary thing? Anybody? Z-scheme is a sequential serial connectivity. Will it justify? Will it justify? No. Only parallel connectivity can justify enhanced oxygen production. All right? Things like that. Anyway, so you people should say, now, when I was in Mala, I was a teacher in Mala, there was a girl in Rajgopal's class. Where is Rajgopal? Yes. Nita. One day we were sitting and discussing, and she came to me and said, Sir, ഇതൊക്കെ ഇങ്ങനെ നടക്കുവോ? ഇത് ഇവിടുന്ന് ഇങ്ങോട്ട് പോണു, അവിടുന്ന് ഇങ്ങോട്ട് വരുന്നു. ഇത് വെള്ളരിക്കപ്പട്ടണം പോലെയാണല്ലോ. Exactly, that's how I felt when I studied that. Exactly. That's how I felt. So I thought all this had to go. I sat down and studied, came up with a better explanation. All right. So if you have genuine doubts, learn to, because that girl also came and said to me, I also realized, yeah, if I also felt, she also felt, it is not commonsensical. And then this logic, two things give greater current or electron flow. It can only be possible with parallel connectivity. Simple things. So like that. Now, let me take you to the next part. The speakers of these talks were somebody different. One guy has gone. Now, have you seen this
bacterial flagella problem. You know that bacteria have flagella? These were quickly put up. So now the classical theory says bacterial flagella rotate. This is the structure. All right. I have some video. A video में काट क्यों? 1 second before that. So this is the structure of the bacterial flagella, and there are only 2 rotary structures known. Both rotary structures our work has completely demolished. There was a scientist called Michael Behe. He said that rotary structures are irreducibly complex. In 1980, he wrote a book. Why? To make this protein work, 7 distinct phases this thing will have to go through. It has to pierce. Have you seen any rotary structures auto-assembling? Any rotary structure? No. To get something rotate, say for example a fan, you know, there is bush, all that, the assembly, it's an electromagnetic system where you can have bushes and rings and all that. But those kind of things to get in a natural system, it's impossible. Impossible. So this is the structure. You have a huge one, multiple protein here, and then there is a rod-like thing that goes out. So this is the dimension. When you study biology, please study dimensions. See if the dimensions fit. See if the forces agree. All those kinds of physical feel is very essential if you're studying biology. Otherwise, you will end up saying 4 elephants can sit in a Maruti car. That's what. All right. So you are supposed to know the dimensions and the forces at very sub-micron kind of level. That's what biologists are supposed to do. If you don't do that, it's very difficult. So rotation, it's very difficult. And this is supposed to rotate counterclockwise as well as clockwise. So it's supposed to be like this. If this is the piercing rod through the membranes and cell wall, this rotates at around, this big one rotates around 2000 RPM. 2000 RPM. If this rotates up at 2000 RPM, earlier this was supposed to be the primary rotor, but then they found that the idea was proton based. So when they mutated and found that nothing happens, then they said there was another MOT-B protein here. They said this rotates. So if this rotates at 2000 by the principles of gearing, this should rotate something around 13,000 or something, that high number. So not only rotate in that number, it can switch from clockwise to counterclockwise. And that was through some way of gear switching. Such sophisticated materials and mechanisms in bacteria were advocated. This is the structure. This is flea N, flea M, flea G. The name, this is MOT AB, flea F. And this is the rod. This is the export apparatus. So if the flagella is made of flagellins, monomers, it's made of one monomer. The monomer goes through this and gets assembled. So there is the cytoplasm, cell membrane, periplasm, external barrier. All right. So this is the rotor and this is the motor, stator earlier. But now this has become also a motor. Earlier they changed it. Okay. Now I wanted to show you that. Can you? There's a video online, a 2017 fluid dynamics conference by yes, please see, this doesn't have any audio. See this. Many bacteria propel themselves by rotating thin filaments like this. This is what it is. And this is about a micron dimension. This is what you can see microscopically. Okay. Now, the flagella are approximately 20 nanometer in thickness. So the light that you can use, the lasers that you can use to visualize through fluorescence and all that, they are 200, or maximum resolution that you can get is 100 to 50 nanometers. The dimension is 20 nanometer crossed. So this rotation you can never see. Okay. So now what they did is, this flagellin they disassembled and assembled in lab, and then they stuck it on a film. And when the water flowed, you can see how that spiral flagella, now you see the water is flowing like this. It looks like rotating. Is that possible? If it is stuck at one end, can it rotate? And this doesn't have the basal assembly. Now this is just stuck. It doesn't have the basal assembly of rotor and motor and all that. This is just the filament. The filament is spiral. If it is stuck on the slide, the more you get, it looks like rotating. See all that. There is no motor, there is no rotor, it's just stuck. The material, when the water flows, it looks like it is rotating. Did you understand this part? So clearly, it is not rotation. We piece together that it is a precession. What is a precession? Now, if the water comes out here, I will show you what our theory is. That's enough. That's enough. Thank you. Can you put the other slide back, please? Thank you. Now, for me, if I was in the field, if I saw this, I would immediately throw out the rotary model. Because it's an optical illusion. Very clear that it's an optical illusion. I have shown you so many experimental observations, basic criteria, which a 10th class, 12th class student could have seen, questioned, and used to overthrow. So rotation, revolution, spiral movement, torsional, all these are very different. So if a rod has to move, advection, direct torque, gearing, belting, these are the different ones. The natural way of, without having any problem of continuous 360 rotation, is only possible by human-made, magnetic field incorporating systems. Mechanical systems, very difficult like that. So here is one beautiful thing. They're all type 3 secreting systems, the bacterial flagellar system. What is a secreting system? Something that pushes some internal component outward. So you see, type 3, type 4, this is the secretory system, and it is used to push some internal fluid out, toxins out, all that. So in the Murburn model, yet another aspect, it is a secretory system. It is not a rotary system. Now, yet another important thing, Howard Berg from Caltech, Harvard, and later he was in these two instances, he found this so-called rotation, and he said it's he identified the earlier structures, said that it's a left-hand spiral. Now, if you take a left-hand spiral, and they said that it's a counterclockwise movement. If a left-hand spiral goes through a counterclockwise movement, it can, now this is the bacterium, if this is the flagellum, if this is a left-hand spiral, then if it goes through counterclockwise movement, then it will pull the bacterium through normal hydrodynamic basics, and it will not push. So anyway, the basic argument that they use cannot work. The most important thing is, this is a very important biophysics problem, because it's a low Reynolds number, something that he was talking about. At this dimension, the inertial forces don't work. Here, you have to think of some other way. you know. So that's why they had this big ideas. And then the mechanism of bacterial flagellar motor was not based on ATP, but it was based on proton gradient. Somehow the protons were supposed to come in like this. Protons came in, they set this going round and round. These kuti things set this thing round and round, and this long thing went round and round. So propel like that. Now, the point is, and then uncouplers, they said that uncouplers were the reasons that showed protons were very important. Now, the problem is there are many molecules that don't have protons, but they also uncouple, so as to speak of. How? So it's not proton based. So if a proton can't go up and down a membrane on its own, something charged, nitro, 1 plus, 1 minus, plus, minus, these kind of molecules, how can they go like that? Just because some people said they bought it. Proton is a very small unit charge. If it cannot go from here to there, how can these molecules with multiple charges, how can they go so quickly? That's another basic. What happened? Sorry. Yeah. So you see, this is the structure of bacterial flagella. Now you see they have grooves from the base connecting to outside like this. The structure is like this, very beautiful. And not that is prokaryote, that is ordinary bacteria. This is archaebacteria. They also have these grooves kind of, you see. Now, this is the classical rotary model. It says left-hand screw. We say that is not possible. The Murburn model says metabolic water is pushed out and it goes through the grooves. And this one behaves like a elastohydrodynamic system. This filament is elastic and it vibrates, and that vibration passes on to the remaining part. And the one, it looks like going around. Now it can explain how you stick it one place without rotor, without motor. It can. So it's an optical illusion, this round going round and round. So there is a, this was an earlier slide. It's not actually a thrust. It is. Now you imagine if water is just thrown out, it doesn't work. So I figured out that this part is elastohydrodynamic. Okay, so then you pinning a cue. Sorry. Sorry. So the Murburn model does not have both counterclockwise and clockwise. It has only counterclockwise. It doesn't say this. We say the apparent rotation is a optical illusion, and there is only counterclockwise precision that is afforded. So here you see the molecule, here if you see they have high amounts of polar and positively charged residues. So they can easily convert ROS into water and also recruit water there. So one very big thing is bacteria can even swim at pH 11, 12 alkaline state. So where is the proton there? So what they said, it's not proton there, it is sodium. Now you imagine where is one proton, what is one sodium? Very different ion. Like I said earlier, proton is 0.00002 angstrom, whereas sodium is 2 angstroms or more. All right. So how can the same machinery, and now you see the structures. Oh, these are the structures here. The structures are not given here in this slide. I think I put it together in the last moment. I'm sorry for that. The biggest part is they said there is a one amino acid D32 conserved in this MOT. This is the MOT. There is MOT AB. I should have put the bigger image. This doesn't have it. So what happened is that is conserved. Now we checked, Danny checked the conservation and saw D32 is conserved. We found so many examples where there are no D or E. There are no D or E conservation. So what were all these people who were studying this field doing? Whoever has money, study. That's what they were doing. We have explored 20-25 different systems. We feel, oh, this is an interesting problem, we come there. There are many researchers working on this thing for decades altogether. If they say this is conserved, they should have seen the conservation. They didn't. Somebody said it is conserved, so it's not conserved. And there are many systems where there are multiple acidic residues. Then, if this is such a beautiful mechanically conserved system with gears and levers, you would imagine that their structures, the number of amino acids, all that will also be similar. There is no conservation whatsoever. The lengths, the types, everything is different. So, classical model of bacterial flagellar motility has proton motive force, MOT AB has status rotating the steering, rigid rotating flagellum, propeller like swimming. It uses proton motive force, rotor, hook, rotating filament, and swimming. Questions? Not enough proton. One mitochondria, it's just like a mitochondria. Bacterial body does not have enough protons. Then million type RPM nanomotor, how can that be possible? Direct proof. There is no direct proof. No unified explanation of motility. Energetically not possible. So what Murburn model says is that when you do Murburn for one molecule of glucose with Murburn kind of oxidation, you get 36 molecules of water formed. If you study, analyze. So metabolism produces a lot of water. So the water is extruded from that base of that flagella, and that creates a shear kind of force. It creates a precessing wave, and that by the phenomena of electrohydrodynamics, it gives thrust. All right. Metabolic water generated by redox chemistry drives shear induced bending, no rotary motor. All right. So this is the basic logic. So classical is PMF, rigid rotation, ion gradient, propeller flagella. This is metabolic water, precessing bending, metabolism, electrohydrodynamic, multiple motility. It can explain very many different times. I'm winding up. 4 o'clock, right? You have to get from that UNS. Yes. So classical proton-powered rotary motor, Murburn redox metabolism produces water. So once again, so then biodiversity, ecology, dynamics. Now, why do we produce diversity? Fundamentally, when you have reactive species spontaneously interacting. So Murburn is at a core bottom level, building blocks. They interact like electrostatic, reactive species, whatever. They give catalysis, conservation. And then from there, central dogma and cell theory came. Then you have aerobic and anaerobic systems. So from chaos through time, you can get deterministic order before affinity-based mandates could set in. So foundation, this is the foundation of spectra of genes and species and ecosystems. Murburn, allows distributed redox charge dynamics, energetic decentralization, microenvironmental niches. Just now we got a work done, which explains why microaerobic systems have greater diversity, because different redox regimes allow different kinds of metabolic stabilizations. So that's the reason. So molecular diversity from safe randomness, ecosystem level of integration. Then biodiversity, traditional explain is primarily through random genetic mutations. Add stochastic biochemical variations from redox reactions. Mutations are sponsored by Murburn also. Slow over generations. Here within a generation also you can have rapid changes. Then roles of genetic DNA is a direct blueprint. DNA sets the stage, but Murburn dynamics influence the real-time performance of the phenotype. Level of action, always focusing on genes and proteins. System level focus comes from Murburn. So the biodiversity is an emergent property of redox flexibility and stochastic control in biological energy transduction. It transforms the understanding of the biodiversity from being a byproduct of random evolution to being a necessity or necessary obvious feature of Murburn powered life. So, lignocellulosics, their breakdown, an enzyme cannot go into such complex structures. Only diffusible radical species can go and break down. So carbon cycling, the natural formed halogenics, chloroperoxidase, 95% of naturally formed halogenics before mankind came, it was by enzyme actions featured by Murzymes. So competition, symbiosis, succession, nutrient cycling, systemic health, everything now you can see Murburn contribution. So enables flexible metabolism and energy transduction at a micro scale, at a macro scale, links all living and non-living systems via diffusible redox couplings. Ecological scale explains nutrient cycling, adaptability, dynamic equilibrium. Planetary scales describes as self-organized, non-equilibrium energy network sustaining life. Ecology is the emergent behavior of countless Murburn interactions distributed across scales, from molecules to biospheres. So this is the kind of idea that I wanted to give you. Basically, at multiple levels, you have Murburn working. It explains why you have DNA structured like this, why the bases are tucked in, and you have ribose and phosphate backbone on sites. You want to save it, the hydroxy groups and phosphate groups repel ROS, negatively charged oxygen superoxide. So that way you can explain. Lipids, there is in the lipids, you have less availability of functionalization. So radical species will just kind of have a lifetime there. There is no proton to get converted quickly. So there are certain questions we will take. I just wanted to give you a short overview of the 2 days multiple sessions. What is the most salient feature of life? A living being has the ability to preserve itself by commissioning non-spontaneous reactions. This is a physical perspective. What is the most essential requisite for the above? A simple powering modality interconnected with coherence, homeostasis, and response principles. Does the cell theory or central dogma provide a tangible means for achieving the above non-intelligently and short time scales? No. Murburn concept addresses the above need and complements the cell theory and central dogma as yet another fundamental principle of life. So what is the etiology expansion of Murburn concept? I hope all of you know. Muod burning or molecule unbound ion radical interactions or mild unrestricted redox catalysis. How is Murburn concept relevant to life? Murburn concept conceives cell as a simple chemical engine performing useful work via the principle of effective charge separation brought about by the obligatory involvement of diffusible reactive species or multiple things that Alan expanded perhaps from the interactive chemo-electromagnetic matrix. So it sets in a matrix, electrostatic, diffusible species, all that it sets in a matrix. Movement of one thing here perturbs the other. So that's how everything gets connected.
And from there, you emergence of biological intelligence. It's not that a membrane protein can pump 3 sodium out, pump 2 potassium in. All that intelligence can never happen. I can bet my life on it. It is impossible. And there is no evidence for it also. Why is Murburn concept a fundamental principle of life? Murburn concept provides a non-intelligent and stochastic framework of interactions between diverse molecules and ions that constitute the cell. In doing so, it enables a simple explanation for powering, homeostasis, coherence, signal transduction, electromechanical activities, etc. So last slide. What is the salient difference between classical and Murburn? Classical perceptions primarily deem DRS as toxic waste products, whereas Murburn school deems it as an essential elixir of life. Does Murburn concept oppose or negate classical perceptions? Murburn concept is a larger set of ideas, including several aspects of the classical views, incorporating later known facts and improving upon earlier prevailing stigmatic or aesthetic notions. Stigmatic and aesthetic notions mean, oh, reactive oxygen species are bad, only bad. So in quantitative perspective, what is the impact of Murburn concept? Classical perceptions are purely deterministic schemes to account for metabolic or physiological outcomes, thereby mandating integral stoichiometry, whereas Murburn concept accommodates stochastic events, thereby explaining non-integral stoichiometry in cellular process as contributors of cellular function. So with this, the formal lectures come to an end. It's already 4 o'clock. The organizers want us to end quickly, and the crowd has also thinned out. Yeah, online. Yeah, Yanyuvu, sorry, Mo, can you? Mr. Mo, are you there? Oh, he has left. Yeah, he has left. So he has left. So no problem. So anybody wants to say anything at this juncture? Chairs, please.
Sir, you have a very nice presentation, but the ecological, and you have studied, explained the details about regarding the Murburn concept with the that is the mechanism of bacterial flagella motor as well as the photosynthesis in a scientific way. But I think more studies are required to establish the fact as a whole.
Yes, madam. You from 2017 from 1999, I have been working on this. The fact that photosynthesis, with due respect, we have just communicated that frontiers. I have four theoretically wrong ideas, 4 experimentally wrong interpreted ideas, and 4, what was the other aspect? 4. I gave you that yesterday. Anyway, there were 4. If you can take it, madam, and give me even refutation to one idea, experimental observations discounted, all that. Basically, what has happened is the consensus has prevailed. Daniel Arnon, who, if you remember, cyclic photosynthesis, those reactions, when he differed with the mainstream and he said that, oh, NADPH can be formed even at photosystem 1. It need not go through the Z scheme. They silenced it. Bicarbonate, very clearly the data clearly refutes photosynthesis, Z scheme. So if you care, madam, my talk is available on YouTube. It elaborates, and we have published around 10 papers on photosynthesis. So it has systematically studied 2 papers published in JBSD. Systematically, all published data we have accounted for. We have refuted 45 points to say why the classical explanation is wrong. So in my philosophy, I can accept that Murburn model can be wrong, but I will assert, let this be wrong, but I will assert that Z scheme is definitely wrong. Why? The explanation for the Emerson experiment to explain higher yield, how can you put it in series? That's evidently wrong. Things like that, So things that don't, the
20% you get ATP with just rhodopsin. How can rhodopsin give? So rhodopsin can make reactive oxygen species. It is not a proton pump. Rhodopsin has got carotenoid. It is well-known chemistry. When carotenoid is hit by light, it produces reactive oxygen species, ROS. So I have been doing this work for about 27, 28 years now. I have conclusively shown that the classical theory, what we teach in school and colleges and in universities, is wrong without a doubt. Without a doubt, man. Without a doubt. I can bet my life on it. It is wrong. I can also accept that Murburn is wrong. Because there could be something wrong. But for sure, as of now, I don't know what is the wrong thing about Murburn model. If you say this is not right about Murburn model, and if it appeals me, I'll jettison in that moment itself. But we have got enough arguments and evidence, theoretical, experimental, all that to show that the classical J-scheme is wrong. Koch-Jolio cycle is wrong. Q cycle is wrong. These are all part of the redundant idea. It is published, that material. In fact, my preprint assessing Z-scheme as an explanation, it has got around 5,000 odd downloads in Open Science Foundation preprint, madam. And now I used to put my preprints there. They closed that preprint also for life science on. So now we have to go through all this. Only now I have ArcSiv and BioArcSiv and some other journals. So I was getting there were followers for my work, and that's a modality for them to disconnect me from my followers. So my paper in Biochemistry Insights, 10,000 downloads, most widely read. They discontinued that journal. So what to do? So I'm very sure that the classical theory is wrong, but my theory also could be wrong. If you find some fault, I'll be very happy. It will help me go to the right one. Yes, madam. Thank you.
Okay, all the very best for your
Thank you, madam.
enthusiasm.
Thank you, madam.
Thank you. It was an excellent, for me, new information about this concept and about photosynthesis. In fact, we need to go thoroughly. Here, wish you all the best. Hope it will take some more time to accept this idea by the people.
I wanted to share one video, if possible, to see the response. Is it possible to check the net now? Is it possible? Just 1 second. Can you check the net? My channel, the man who wrote textbook photosynthesis for the last 20 years. Satyam Jaitu channel, YouTube. 2 minutes. 2 minutes. 2 minutes. Okay. Satyam Jaitu. That's my channel. Satyam Jaitu. Just one. This one. In 2019, please play this video. Yeah, this one.
Wait, how old is the new hire?
I don't know, but he literally.
In Kalidas, something I forget, I'm not very well versed, that he sat on the tree trunk and cut himself so he falls. So I don't want you to fall. I want you to be successful. But you please listen to reason, way of presenting, the way of writing. If you constantly attack, you are attacking yourself by doing that.
Right. Something that now you should go from your presentation to the primary.
Please listen to it carefully.
Where you publish this? Now that is not published.
Yeah, but if you run the forum, you can seminar, you say, well, of course everybody knows that you
now know, but until 4 years back.
So probably why did I put that? Because you keep on saying that there is wrong. It's controversial. Then ask me if it's controversial. If it's controversial because of you. What I'm telling you. Because you don't know how to present it. You don't know how to write it. You don't know how to do it. You have to use simple. I even read the abstract. 10 words are there. So you want those people who know tough English, they're bombastic English, they don't understand. Not a simple-minded thing so that the common man, publish it in nature. And your work is that it should be published there. But you are not working in that end, you are working opposite. You are killing yourself by not listening to see how you can present your talk, how you can write your paper, so that people will understand your theory. I'm sorry, as I said, take my PhD, take my professorship away, because I mean well. I know you didn't say that, I'm saying that. I mean well for you, and I believe in you. That's why I wanted you to be here. But you disappointed me by not explaining your theory from the step one. I've never ever said that in my life to anyone.
I think it's a matter of in India we don't question. I'm glad you're coming by you because that's about what you people question the way that.
As I said, I've taught for 40 years and I'm listening to speeches after speeches and talks after talks since 1953.
But
two Rudy Marcus who came to Ghana. I know. Explain the concept. I've given you the papers. No, that's another thing. That is again very wrong for you. I've given the papers, God damn it, you read every word and you should understand. No, you should be able to write in a way that people understand. A man can say, don't explain, because this is a problem. It continues to be a problem. So unless you learn that you have a brilliant idea and you have to learn sit down to write it in a way that people will understand step by step.
The thing is, it's not a tangent which. The thing is that we should not be aggressive about attacking.
What more aggressive? That your word.
8 minutes. This is the important place, huh? This is the very important place. This part is, yes. Yeah. What happened? Sir?
In a way that people will understand step by step.
The thing is, it's not a tangent which. The thing is that we should not be aggressive about attacking.
What more aggressive? That your word. Even when you're talking to them, and now what do you say? This is not the way to speak to a group of young people.
They will work on the.
You need to learn. And if you don't learn, well, you're running wrong. Let's see. Yeah, well, you are very, what's the next thing?
Yes.
Arrogance at the highest level. No. Interested in your life, only interested. I believe in you. Everybody says, Govinji, are you crazy? You're going to listen to Murlis? No. Because I thought I'd come here and you'll explain.
ChatGPT सीखना है? आ जाओ, सिखा दूंगा.
They know my work. They know my work. He says, are you crazy? You're going to listen to Murli's work. The work is published. It's out there. They pretend that they don't know it. Century, generations after generations, we teach wrong things. That's not right. When Newton's corpuscular theory was very popular, Huygens proposed the wavefront theory. It took 200 years almost. 200 years. Just because there is somebody powerful, why should people not listen to arguments and evidence? Science is based in claims, arguments, evidence. Claims, arguments, evidence. That's it. Believe in it, young kids. You should do that. We have one more professor, what's his name? KV Venkatesh, star professor from IIT Mumbai. He is supposed to join in. I hope he will join. He and Dr. Grinson, they are the validation, valedictory guests. So are we going to valedictory now, madam? Sir? We'll continue. So can I just call him? I said around 5 o'clock. It's only 4:20 now. Yes, okay, sir. Just 1 second. I'll just call him. He said he'll. Professor KV Venkatesh has got a company called MedFlux, and he's the biggest persona in systems biology in India. So he has agreed to come and speak a few words. KV Venkatesh. I'll call him. I hope he picks my call. He keeps his promises. He is not picking the call. I think then Grinson sir can, 1 second, once more. Yeah, sir, please go ahead. I'll try to reach him. Today morning, he assured me he'll join. Yeah, let us, because 5 means event. Anyway, we will, because most of the people then left us, right? They yes, also want to like that. I understand. So you try, please, and meanwhile, we will start. As far as Swadeshi Science Movement is concerned, it is a very successful and a very novel program we conducted. At least some of you started working on this line, so we'll be very happy. And already a few programs on this line, we have got suggestion from the experts, and we will be going ahead with that. So I also request you to follow Swadeshi Science Movement Kerala programs and forthcoming Science Congress. 33rd Swadeshi Science Congress will be held in a few months from now. And in September, there will be a Malayalam Shastra Sammelan. So every papers will be in Malayalam. So that also scheduled to be conducted in CIFT, ICR Central Institute of Fisheries Technology, Kochi. So these are our forthcoming events. So with this small our activities, I request Dr. Grinson George to conclude this.
Oh, actually, usually the valedictory comes with a proper structured agenda, but here it's more or less like an informal thing. We have been here for 2 days, so just a few of you can say how it went, how was it, what you learned, some feedback, at least from different age group.
From Kerala Veterinary and Nanosciences University, working as a professor in the Department of Pharmacology and Toxicology. So this idea was very, actually I was telling that it was a very new idea to me, very new idea and very concept. But actually I felt like there should be an idea to fill the gap that we find in the research. So I was seeking so many things, even I've gone up to the quantum, which especially when the photosynthesis was there, because there are, I found some lacunae, because without saying, because photosynthesis and direct metabolism, they have some similarities. So the same similarities I found in photosynthesis also. So, I think in 2011, there is some published paper saying that is basically the quantum entanglement and the quantum superpositions that is happening, because that is why the photosynthesis is highly efficient than the conventional cycle. Then regarding the motor proteins, that also, in our research itself, we have found some problems, especially with the direct metabolism, that that I was telling also. When we do it in the real animals, we get the lacunae in the research. Anyway, this was a new concept for me to think of, because this was not known to me. I came as a fresher for this idea, and really this was a thing that I can work out in the real-time experiments and changing the ideas. In our applied research, we don't mind to change the ideas, because when I teach, after 5 years, I will tell up to the past 5 years what I taught is wrong. This is the new mechanism of action. So we and our students, we are used to it, used to change.
That's wonderful. That's wonderful. That's the spirit. That's the spirit.
Accept the changes, because our subject, that is our pharmacology, that changes every time. After 5 years, say maybe 100 drugs will be discovered and 50 drugs will be discontinued due to toxicity or the things. So adapting the changes is not an issue for us, especially when it is coming to the professional courses. Yeah, applied research. We do more with applied research. So that's why I think I can incorporate.
Wonderful. Great. So can we, sir, Professor KV Venkatesh is a star professor from IIT Mumbai, if I may say that. and he's a big name in systems biology, the fields where I talk about. And he has a company called MedFlux, which deals precisely with these kind of things. He was the convener of the first Murburn Concept workshop at IIT Mumbai in 2023. And he also joined us for the event that we conducted on Murburn Concept at Amrita in 2024. And I'm lucky and fortunate to have him join us for at least the valedictory function today. Thank you so much, sir. Please, the floor is yours. Sir, you're on mute. You're on mute.
You're all able to hear? You're all able to hear?
Yes, sir. Yes, sir.
Okay.
Yes, sir. Yes, sir.
Okay.
We can hear you.
Yeah. So it's an honor and privilege that Murli Manoj asked me to deliver this valedictory address in this idea of Murburn concept. I think last 2 days you have had a lot of talk and discussion on this. I've had my contact with Murli, as he was indicating, from last, I think, 7-8 years, and then I've sort of followed up with some of his work. What I felt was, the science advances not through established knowledge, right? So we need to challenge many of these sort of assumptions and come out of the comfort zone. So any transformative ideas begin with such alternative hypotheses, go through rigorous debate, go through experimentation, refinement. I think this Murburn concept is also going through that framework, thought-provoking, but going through that framework. And it always takes time when you have ideas which you need to bring out of the comfort zone. So the very idea that reactive oxygen species, which was considered, only to be stress, aging, disease-related, to give an alternative in terms of having core physiology, thought process in terms of controlling and mediating catalysis, electronic transfer, cellular communication. So, its role in all this is what the Murburn concept sort of brings in, trying to bring in another idea in the biochemistry and biophysics. So it's a conceptual shift. And as I was indicating, any conceptual shift takes time. And then more and more experiments have to be done. More and more people have to prove.
Yeah, go ahead.
Yeah. You're able to hear, right?
Yeah. Yes. For a second, there was a break.
Okay. So what I was telling was such conceptual shift in viewing biology, which is typically, people are comfortable with ordered linear molecular events, catalysis occurring with enzyme active sites, and then shifting to more dynamic microenvironments, with diffusible molecules, and then creating a stochastic zone at which reactive contributions happen in terms of biochemical transformation is quite novel, and then it takes time to get into these. Although in the last couple of years, I think Murburn framework has been used to demonstrate diverse biological phenomena, mainly in oxidative phosphorylation, starting with oxidative phosphorylation. Now, I think in photosynthesis, redox-dependent physiological processes, etc. It may be debated, and people can come up with alternatives, but I think this is very essential that, for long-standing perspective to be had, more and more people should engage, and especially people who get motivated in such processes to involve in their own research and try to add, and expand on this scientific community, which aligns in this sort of thought process. And my own research, we have looked at signaling pathways, immune responses, metabolism, mitochondrial communication, and I see that ROS and this concept sort of fits in. Although we look at a more systems view, but we see that there is this greater and greater complexity that we see. The ROS playing a fundamental role in deciding physiological state becomes central. So to get this broader perspective in my own research as systems biology, I know we go from a reductionist approach to more, sort of a accounting for all the systems interplay, and biological functions essentially emerge not from individual molecular action alone, but from networks, interactions spanning genes, proteins, tissues, organs, and their surrounding environment. So the emergent properties that come because of this interconnected network, from metabolism, immunity, hormonal, now microbiota and environmental influences, where oxidative stress and ROS plays an important role in all of this. And to put a framework where, at right amounts, they support physiology, and at some abnormal levels, they become toxic, is fundamental to Murburn concept, which also aligns with the systems physiology that I myself have been sort of looking at. So whether one would fully agree with this concept or not is a different aspect, but it is an important scientific principle, and it fits into biology being dynamic, interconnected, and multidimensional. And therefore, I'm happy that these kinds of conversations and then conferences are held where we look at the concepts in a different viewpoint. And as I was indicating, this fits into my own thought process of systems biology. So I don't want to, sort of, I'm sure the audience have been, enriched by various speakers on aspects of Murburn concept and other, biophysical and biochemical concepts. But as this concludes, I just want to say that every scientific idea has value, when it stimulates curiosity, it encourages dialogues, and then it inspires better experiments and testing out new ideas. So innovation always flourishes when both this intellectual courage to question and propose new ideas and a scientific discipline to prove to a point that they're rigorously tested. These two should continuously to happen. So I want to conclude with saying that such a thought process has to be cultivated. A culture has to be cultivated where questioning is welcomed, evidences are respected, collaborations are encouraged. And then going forward, I think with a system thinking guidelines, the new discovery should happen. So I thank you, especially, for giving me this opportunity. Murli, thank you for giving me this opportunity to give the valedictory function, to address this valedictory function. And I hope that you will have more such conferences. And I wish everyone continued success in their advancing science and research. Thank you. Hello. I hope you were able to hear.
On the line. So we missed you, sir, for some time. Yeah, please go ahead, sir.
No, it's okay.
Yes, sir. Yes, please.
Yeah, no, I just wanted to say that, I don't know where I sort of...
last 1 minute, maybe. Last 1 minute, maybe.
Oh, okay. So no, I just wanted to tell that, the future of biomedical science is sort of building, physiological understanding, connecting molecular mechanisms to the whole organism function, for identification of new therapeutics, maybe, somebody was talking about, novel drugs and all that, precision medicine, rational design. So that is where it is heading. So new concepts have to come, like Murburn concept, new concepts have to come if you want to bring in these kind of meaningful mechanistic knowledge for innovative ideas. So just to, as we conclude, I just want to remind that every scientific idea has value when it, it stimulates curiosity. It has to encourage dialogue and inspire better experiments. People have to have the intellectual courage to question many things, propose new ideas, but respect the scientific discipline for its rigor, to go through rigor. Okay, so the scientific rigor has to be there. So I conclude by saying that we need to cultivate this culture of questioning and respect the evidence, have more collaborations and dialogues, and then moving forward, have a systems thinking to look at new discoveries. So I thank Murli for giving me this opportunity, and I wish everyone a successful in their advancing the science towards benefiting the humanity. Yeah, thank you.
Yes, sir. Sir, do you want to ask him? Okay. Does anybody want to ask Professor KV Venkatesh? He's a very well-known figure in this field. Dr. Grinson George is also the other person on the floor, sir. He's the director of this institute. Then we have the former president of SSKM, Dr. Muralidharan, also here. He's been the organizer of this event with the Swadeshi Science Movement Kerala. So,
okay, excellent.
Yeah. Please.
Okay, so we were taking the feedback. Professor Suresh spoke and... Yes, sir. If you can, if you have time, you can continue being with us online, sir.
Yeah.
We are just concluding the meeting with this. We'll take some
feedback. Thanks a lot for giving me this opportunity. And I hope that someday I will visit your institute and then, interact and collaborate with the faculty there. Yeah, I'm looking forward to that. Yeah.
It's a pleasure, sir. We are a 80-year-old institute based at Kochi. It comes under Indian Council of Agriculture Research. It's Indian Council of Agriculture Research, Central Marine Fisheries Research Institute. This is the biggest research institute in Indian Council of Agriculture Research other than deemed universities. So education is taken care of by the deemed universities. We deal with whatever people think that it is the geographical boundary of India. We start from there and we go up to the exclusive economic zone. So that
Acha.
resources we work on. So
Sure,
we can,
sure. Yeah.
headquarters is at Cochin. We have a center in Mumbai also. That's
Oh, okay. Yeah, yeah. Okay, thanks a lot. Yeah. Take care.
Sure.
Bye.
Thank you, sir.
Yeah, bye.
So any more feedbacks? Younger age groups? The youngest of you can tell. There should be a claim for the youngest, no, otherwise I have to speak. Need not wait for the youngest also. If you were somewhere in that category, that length frequency or age frequency class, you can come and speak.
Say something, kiddos.
There are no feedbacks.
No feedback.
No, we need not restrict to the younger group. The others also can, if they want something to say. Just to know how it went. What was the
Kochi sir, Parayu, Negilu. He's in his 70s. He has come. He's
Hello. ഇത് സയന്റിഫിക് ടെക്നിക്സ് ഒന്നും എനിക്ക് അറിയില്ല. പക്ഷെ എനിക്ക് മനസ്സിലാകുന്ന കുറെ കാര്യങ്ങൾ എന്ന് പറയുന്നത് ആത്മീയമായിട്ടുള്ള ചില പ്രശ്നങ്ങളാണ്. സ്പിരിച്വൽ ആത്മീയത എന്ന് പറയുമ്പോൾ നമ്മൾ സാധാരണ ഉദ്ദേശിക്കുന്ന ആത്മീയതയല്ല ഉദ്ദേശിക്കുന്നത്. കമ്മ്യൂണിക്കേഷനാണ് ഇവിടെ ഏറ്റവും വലിയ പ്രശ്നമായിട്ട് എനിക്ക് മനസ്സിലായത്. അതിൽ സാറ് പറഞ്ഞ പോലെ കമ്മ്യൂണിക്കേഷൻ എങ്ങനെ നമുക്ക് കമ്മ്യൂണിക്കേറ്റ്, ഒരു ചെടി മറ്റൊരു ചെടിയോട് കമ്മ്യൂണിക്കേറ്റ് ചെയ്യുന്നുണ്ട് എന്ന് റിസർച്ചുകൾ പറയുന്നുണ്ട്. അതിന്റെ സാങ്കേതികമായ എങ്ങനെ അത് സംഭവിക്കുന്നു എന്നാണ് മനോജ് സാർ പറഞ്ഞതെന്നാണ് എനിക്ക് മനസ്സിലായത്. പിന്നെ ഈ പറഞ്ഞ പോലെ ഈ ചാനൽസ് ആർ ക്രിയേറ്റഡ് ബൈ പ്രോട്ടോൺസ് എന്ന് സാർ അങ്ങനെ ഒരു സംഭവം സാർ പറയുന്നുണ്ടായിരുന്നു. ഇതിനകത്ത് സയന്റിഫിക് ആയിട്ട് പറയുമ്പോൾ അതിനകത്ത് ഇത് പറഞ്ഞു. അതെനിക്ക് എന്താ പറഞ്ഞാൽ അതെനിക്ക് വലിയൊരു സംഭവമായിട്ട് എനിക്ക് തോന്നി. കാരണം ഇടനാഴികൾ സൃഷ്ടിക്കുക, ചാനൽ എന്ന് പറഞ്ഞാൽ ഇടനാഴികളെന്നോ ചാനൽ എന്ന് പറഞ്ഞാൽ അല്ലെങ്കിൽ പോകുന്ന മീഡിയം എന്നൊക്കെയാണല്ലോ. അതിനെക്കുറിച്ചൊക്കെ പറഞ്ഞ ഭാഗങ്ങൾ റിസർച്ച് ചെയ്യാൻ ധാരാളം കാര്യങ്ങളുണ്ട്. അത് ഇന്ത്യൻ സിസ്റ്റത്തിൽ തന്നെ നമുക്കത് കിട്ടുന്നുണ്ട്. മനസ്സിലായോ? എനിക്ക് സ്വതേ ഇപ്പോ ഇതിനകത്ത് കുറച്ച് രാഷ്ട്രീയ ഭാവങ്ങൾ വരുന്നുണ്ട്. അത് പുറത്തും ഉണ്ടായിരുന്നു. റിസർച്ചുകൾ നടക്കുമ്പോൾ എല്ലായിടത്തും രാജ്യങ്ങൾ നോബൽ പ്രൈസ് ഒക്കെ കിട്ടുമ്പോൾ ഈ പ്രശ്നങ്ങൾ ധാരാളം ഉണ്ടായിരുന്നു. നമുക്ക് കിട്ടിയിരുന്നില്ല. ഇപ്പൊ നമ്മളെ ടാക്യോൺസിനെ കുറിച്ചൊക്കെ സംസാരിക്കുമ്പോൾ നമുക്ക് ECG സുദർശനൊക്കെ സംഭവിച്ച കാര്യങ്ങൾ നമുക്ക് അറിയാവുന്നതാണ്. അതിന്റെ വ്യക്തമായിട്ട് നമുക്ക് ഇപ്പോഴും ഐഡിയാസ് ഇല്ല. മനസ്സിലായോ? അതേ സ്ഥലത്താണ് നമ്മളെ ഇന്ത്യൻ സിസ്റ്റത്തില് ഇപ്പോ ഈ സ്വദേശി കോൺഗ്രസ് എന്താ പറഞ്ഞ ഞാൻ കേട്ടിട്ടുള്ള അത്രമാത്രമേ ഉള്ളൂ. ഞാൻ ആദ്യമായിട്ടാണ് ഇങ്ങനെ ഒരു സംഭവത്തിലേക്ക് വരുന്നത്. ഇതിന്റെ താല്പര്യം കൊണ്ടാണ്, പുതിയ വിഷയമാണെന്ന് കണ്ടുകൊണ്ടാണ് വന്നത്. ഇത് നന്നായിരുന്നു. ഇത് പറഞ്ഞല്ലേ വളരെ ശക്തമായിട്ട് തന്നെ ഇത് നമ്മൾ ആ ചിന്തകളിലേക്ക് കൊണ്ടുപോകേണ്ടതാണ്. ഈ സാറുമായിട്ട് എനിക്കുള്ള കമ്മ്യൂണിക്കേറ്റ് ചെയ്യണമെന്നുണ്ട്. അതുകൊണ്ടാണ് സാറിന് ചുറ്റുമുള്ള സാറ് വർക്ക് ചെയ്യുന്ന സ്ഥാപനത്തിലെ ചില ആൾക്കാരെയൊക്കെ എനിക്ക് പരിചയമുണ്ടെന്നും കൂടി ഞാൻ പറയാനിടയായത്. അതൊരു
Okay. I think Murli sir is also showing some impatience. We have to complete that. It's time. So thanks for being with us for you. Yeah. Okay.
The others also, not only director. I had to come again here. The feedback that we can give is that it was really awesome. Thank you.
Okay.
And, we really enjoyed our.
Okay. No,
the arrangement by Ashish. Perfect.
Not to see.
Where is Ashish? Yeah. Impeccable. Very much in the moment. Vision, planning, execution. I can't say there was anything.
There is no much compliment better than what others say. I will always find faults only, but others will compliment. Thank you, Ashish. So
it did not feel that we are in a new place,
alien environment,
an alien place. So it was very, felt very homely.
Okay, thank you.
you for the hospitality.
Thank you for that, Ashish. We'll continue doing that. So thanks for all the good words you have said. And just as conclusion, I want to say there are a lot of videos we see in WhatsApp. So in one video, I was seeing a herd of cattle was moving, and one cattle suddenly jumped, and all the cattle started jumping. Same way, there is another thing which was indicated a group of monkeys were kept in a cages and ladder was given and they were throwing hot water. So initially the monkeys were reluctant to, they were trying to climb, but the hot water was coming, they were not climbing. Then subsequently, nobody was climbing, anybody was climbing, the monkeys who were existing there, they were trying to prevent them from climbing. So then a total new groups of monkeys came, but all were trained in such a way that it became a practice that nobody will try to climb the ladder and try to escape. So same way in science also, when it comes to any administrative decision as technocrats, we also decide based on precedents. We will immediately, if there is a conflict, then we will ask for precedents. What happened before? So that precedents always guide us. That's how we go ahead. And if something which is not in precedents, that takes a longer time to get established. So probably that is happening in the case of Dr. Murali also. But unless and until this precedence is getting established, the people will not accept it. So we have to see how the precedents can be established. Precedents can be established because people have to act on something what is proposed. So you are all going to be brand ambassadors for Dr. Murali. You have to continue working on these lines, flood your ideas in different ways of science, be it an experiment, be it a publication. You have to go on producing more and more in this thought process. Then finally, it will come as an established theory. There will be applications coming out, as Professor Suresh was indicating. Suresh, correct? No? Yes. So Professor Suresh was indicating. So when applications start coming in, people like us, we are basically an institute which works on applied research, not into basic. Some strategic research and mostly applied research. So when it comes to us, there should be a proven technology which can be readily inculcated into an application system. So we are looking for such outputs. One day, probably in this institute, people will be talking on Murburn as a basic concept which has created innovations in marine fishery sector also. So thank you for introducing Dr. Kelod Murali Manoj, who was instrumental in shaping your ideas, thought process for the last 2 days. You may find things which are not digestible. Think on it, why it is not digestible. So either you will find a solution or you will say that, okay, no further research is required in this arena. So a lot of things can come up. Keep asking the questions and try to come up with some solutions which will either take you forward in this line, or you can say that, okay, this is not the right path also. In both ways, it's good. It's good for science. Continue doing at least a thought process to your regular day scientific thinking. So thanks, Dr. Murali, Anitha Madam, and Lada Madam, and others for sitting here, and all the other resource persons. I am not sure who all are resource persons. I could not listen to your lectures. So thank you, all of you. The resource persons might have done a good job, because that's why people are still there. Otherwise, people would have left. I have seen this auditorium where hardly the valedictory speakers are there, and rest all the people have left. But at least half of the students have remained. That indicates that towards the valedictory, that indicates that there is some fire which has been ignited. Take this fire and spread it. Not like the wildfire, but the science fire. Thank you.
Thank
you.
Thank you.
Within 1 minute, I will complete my vote of time. As we know that all these facilities are provided by ICAR Central Marine Fisheries Research Institute under the leadership of Grinson George. So in one word, I am thanking him and all his colleagues who very much supported. Without their support, this would have not happened. So thank you, all of you, including Ashish, and already he got a thanks. Also, I'm not giving it. Then I thank all the students with their, though the classes are there, but they took interest and come forward to attend this program. Also the discussion and those who chaired session, etc., and all the resource persons. And it's really a voluntary work. See, ours is an NGO, Swadeshi Science Movement, and all of us work without any monetary benefit. So, and similarly, all the resource persons came with their own
resources. So we are very much thankful, and without you, this would have not been a successful event also. And many good ideas came, especially our VPN sir told one workshop with St. John's, Bangalore on physics and Murburn, or I put it as a physics, marriage of physics and biology, something like that. And then he is also interested in some drug related things. And like that. So we'll be moving ahead. So I thank also the Amrita Vishwa Vidyapeetham. Many faculties are from there, their academic support. And all of you, once again, thanks, and welcome you to work with the Swadeshi Science Movement in future. Thank
you. Tea is outside.
Yeah, tea is served outside. I did my PhD in physics despite being a biologist. So when Murli sir said my guide was an engineer, but my JRF was in life science.